कैमरा अभी तक स्टार्ट ये नहीं हुआ, रिपेयर नहीं हुआ, कैमरा ऑन नहीं हुआ अभी तक। कैमरा ऑफ है। Good morning, everyone. Good morning.
Good morning, ma'am.
So, how are you all? You all are good?
Yes, ma'am.
Any notification from AIIMS for joining?
No, ma'am.
No.
No, ma'am.
Okay. I hope that we will meet at least during your during your Viva. Okay. So, shall we wait for 1-2 minutes? 83 people are there in the group. So in 2 days we will finish the hematology lectures. Okay. So only we have one tutorial next week. So this is the means it forms a significant portion for the first semester hematology. You have so many practicals on hematology. Okay. Okay, so the today's topic is development function and fate of platelets. Okay, now what is platelets? So till now we have seen different cells which are present in the blood, like granulocytes. Granulocytes. What is their function? Okay. Now let us see about the platelets. So the platelets are nothing but the cell fragments. Even though these are only cell fragments, they perform very important function. They are loaded with granules, okay, with different contents, and they perform very important function of hemostasis. Now hemostasis means stoppage of bleeding. Okay, stasis means stop. Okay, so they will stop the bleeding. So it is very important function which is required for day-to-day life. Okay, otherwise there will be if there is any injury, then there will be continuous bleeding. Okay, and then blood volume will decrease. So so to prevent it and to maintain homeostasis, it is very important function of platelets. Now let us start with the lecture. Okay, so how they are developed. So now you know that so the how platelets develop. So they also develop at the same site where the RBCs and WBCs develop, like in starting from yolk sac, then liver, spleen, then bone marrow. Okay, So in the adult life, thrombopoiesis, it is called as thrombopoiesis because other name for platelet is thrombocyte. Okay, because they are important in the formation of thrombus. Thrombus is the blood clot in the circulation. Okay, so in the blood vessel, so they are called as thrombocytes, and their synthesis or their formation is called as thrombopoiesis. Now they, like other cells, they also originate from the hematopoietic stem cells. Okay, that is multipotent hematopoietic stem cells give rise to megakaryocytes, and megakaryocytes give rise to platelets. So this is hemocytoblast, which is a stem cell which multiply and differentiate into megakaryoblast. Okay, so this is the precursor cell which differentiate into promegakaryocyte, and then any problem? Okay, so this promegakaryocyte then develops into megakaryocyte, and what happened? Any issue? Can you hear me?
Yes, ma'am. Yes, ma'am.
Okay, ठीक है. So then megakaryocytes then give rise to platelets. So these platelets are nothing but the fragments of the cell. So you can see there will be formation of pseudopodia like structures, and then they detach from the cell, and then they form the platelets. Now these platelets, they have the granules inside them. Okay, and these platelets then enter the circulation. Okay, in the peripheral blood smear also we have seen these are the small particle like structures which are present in the circulation. So initially when they are formed, they differ in the size and shape, but after maturity they will become uniform in size and shape. Then okay, so what is the function of these platelets? Okay, so it is the maintenance of endothelial homeostasis, and it will seal up the leaks present in the capillary beds. So maintenance of endothelial homeostasis means endothelial cells secrete some substances like prostacyclin, okay, which causes vasodilation. Now platelet will secrete some substances like thromboxane A2, which causes vasoconstriction. Like this, along with endothelial cells, they are involved in maintenance of homeostasis, and they seal up the leaks which are present in the mostly in the capillary beds. So during day-to-day activity, whenever we apply a pressure on our body part, okay, then there will be damage to the capillaries which are present in the skin and subcutaneous tissue, and these leaks in the capillaries, they are immediately sealed up by the platelets. Okay, if that function is absent, like in dengue fever, when the platelet number becomes very less, then we can observe the petechiae, that is small hemorrhagic spots in the skin and subcutaneous tissue. Okay, that is called as purpura. So that will be observed in patients who have less platelet count. Okay, so normally we don't observe any spots in the skin or subcutaneous tissue because the capillary leaks which happen thousands of times per day, that will be immediately sealed up by these platelets, by the formation of platelet plugs. Okay, so that is the important function of the platelets. Now they also act as the needles of coagulation, and they provide the surface on which the reactions will take place for the coagulation, that is for the activation of different coagulation factors. Okay, and then they will help in the formation of blood clot, that we will discuss in the next lecture. Okay, now and they also assist in wound healing through the delivery and concentration of phospholipids, coagulation factors, growth factors. They secrete various growth factors, okay, and cytokines which help in the healing of the tissue. because they function mostly at the sites where there is a tissue injury. Okay, so because of these growth factors which are present in them, there will be growth of fibrous tissue, connective tissue. Okay, and they will help in the healing of this injured tissue. So these are the small densely packed structures, but they play very important role in the circulatory and defense systems of the human body. They are also involved in defense system by secreting the cytokines. Okay, but here we are going to discuss about only hemostasis. So we have already seen what is thrombopoiesis. So it is developed from the polypotent hematopoietic stem cells. Okay, and these are nothing but the small fragments which are released from this megakaryocyte. Now can you see the nucleus of megakaryocyte? It looks like it is a multi-lobed or multiple nuclei are present in this megakaryocyte. Now let us see what is there, what is present in the nucleus. Okay, so we have two types of precursors for megakaryocytes. So that is colony forming unit megakaryocytes. Okay, so they develop into a simple colonies of 3 to 5 mature megakaryocytes when grown in the culture. And there are another cells which are called as burst forming unit megakaryocyte, and they form large complex colonies with satellite collections of megakaryocytes, and they contain up to several hundred cells. Okay, so it is called as burst forming unit. Okay, so these are the progenitors from which megakaryocytes develop. Now how it is regulated, thrombopoiesis. So the main important factor or regulator for thrombopoiesis is thrombopoietin. Okay, so it stimulates, like we have erythropoietin for erythrocytes. Okay, and this is for thrombocytes, so it is called as thrombopoietin. Now it is the primary regulator, and it promotes the megakaryocyte growth and differentiation, and liver is the main source for this release of thrombopoietin. Now whenever there is decrease in the platelet count, it is how it is regulated, it is based on the platelet count. If there is decrease in the platelet count, within 24 hours there will be increase in the level of the thrombopoietin. So increased level of thrombopoietin will be released by the liver, and then it will start the growth and differentiation of megakaryocytes in the bone marrow, and it will release more number of platelets in the circulation. Now there are other regulators also, like there are some stimulators like KIT ligand. So these in the bone marrow, these immature cells or stem cells have the KIT receptors. Okay, now the KIT receptors are there, and the factors which bind with the KIT receptors, these are called as KIT ligands. Now the stimulators are KIT ligands: interleukin 3, interleukin 1, interleukin 6, interleukin 11, granulocyte colony stimulating factor, granulocyte and monocyte colony stimulating factors. Now you know that these are the interleukins, which interleukin 1 is secreted by macrophage, 3, 6 they are secreted by the helper T cells. Okay, these are also secreted by macrophage and B lymphocytes. So these are the all regulators which are secreted by the immune cells. So whenever there is any injury, or so in case of injury or invasion of bacteria, so all these cytokines are increased and they stimulate the thrombopoiesis. Now we have inhibitors also, because we have to regulate the synthesis of platelets, otherwise their number will increase so much, and it will also leads to increased coagulability of the blood. So there are inhibitors for thrombopoiesis, and these are transforming growth factor alpha, interferon gamma, platelet factor 4. Platelet factor 4 is the important factor which is secreted by platelet itself. So there is this platelet factor 4 will act as a negative feedback mechanism to regulate the thrombopoiesis. Okay, and thromboglobulin. So these are the inhibitors for thrombopoiesis. Now let us see step by step what are the different stages of this thrombopoiesis. So the first, the megakaryoblast is a progenitor. for these platelets, and 19% of the megakaryocyte population is formed by this megakaryoblast. So it has a large, okay, either round or indented lobe nucleus. Now there endomitosis. Now there is one term called as endomitosis which is related to this megakaryoblast. Now this is the peculiar characteristic of megakaryocytes, that they have multiple copies of DNA in their nucleus. Like during maturation, the megakaryocytes grow in size and replicate its DNA, but there will not be any cell division, okay, without cytokinesis, and this process is called as endomitosis. So the DNA will go on replicating. So normally what happens after replication, that there will be division of the cell, okay, but in this case there will not be any division of the cell. So the nucleus of the megakaryocyte will have up to 32 copies of DNA, okay. So that is because of this, the nucleus also has the large and multi-lobed shape. It appears that it has multiple nuclei, megakaryocyte, okay, and that is called as endomitosis. Now cytoplasm is scanty and few or no alpha granules. Now why there is endomitosis, we don't know the reason behind this, okay, but this happens in the megakaryocytes or megakaryoblast. So they are in the megakaryoblast, there will be either no or few alpha granules will be present. Now develop from megakaryoblast, the cell differentiate and it will form the promegakaryocyte. Now 25% of megakaryocyte population is because of this promegakaryocyte, and it has large lobulated nucleus, okay. Cytoplasm increases in amount and the number of granules will also increase. Now alpha granules, what we will see about what are the types of granules which are present in the platelets. Now we have the next stage is granular megakaryocyte. So 56% of population is formed by this granular megakaryocyte. It has lobulated nucleus and cytoplasm is increased in amount and it has abundant granules, okay. Now comes the main step, that is how these platelets will form from this megakaryocyte. So these are the giant cells with multiple copies of DNA in the nucleus. They have large amount of endoplasmic reticulum, okay, smooth endoplasmic reticulum is there. So from this from the plasma membrane of this megakaryocyte, there will be formation of pseudopodia and they will break down into and to form the platelets. So what the platelets will have, the granules which are present in the cytoplasm, and also this endoplasmic reticulum, Golgi apparatus, this cell organelles will also be present in the platelets and mitochondria also, okay. So the whatever present in the cytoplasm, they will enter into these cell fragments and then they will circulate in the tissue. So as they are anuclear, okay, their lifespan is very limited, only up to 7 to 10 days. After 7 to 10 days, they will be phagocytosed by the reticuloendothelial cells. Okay, so let us see about the platelets. So these are small unnucleated fragments with the presence of reddish granules. Their size is very small, it is only 2 micrometers and volume is very low, it is around 8 femtoliters. So you know for femtoliter, this unit is used for RBCs also, okay. And in the RBCs it is around 80 femtoliters. So the volume of platelet is also very less, it is 10 times less than the RBCs, so it is only around 8 femtoliters. And young platelets are in circulation, they are reticulated because they have the reticulum like ribosomes and all the structures in the cytoplasm, okay. So when we stain with the platelets, we can see the reticulum like structure in the cytoplasm. Now how the platelets are, what is the chemical composition of the platelets? So they are formed by the 60% of proteins. Okay, so we have the granular content is mostly protein, has the proteins in it. So 60% of the platelets are formed by the proteins, 15% by the lipids, 8% by the carbohydrates, and they also have others minerals and vitamins. So how these platelets look like under the electron microscope? So the resting there are two phases, resting phase and activated phase. Okay, so during resting phase there will be baseline metabolic activity and they are smooth surfaced, okay, smooth and discoid shaped structure, disc like structures. Okay, and when they get activated, then they will form the size also increases and they will throw multiple pseudopodia. Okay, that we will see in the next lecture, the activated form of platelets, that pseudopodia also called as philopodia. And the platelets which are present, which are circulating in the circulation, they are flat disc with smooth contours. Okay, that smooth contour is very important, it will prevent attachment of platelets with the endothelium or any other tissue. Okay, now they have rare spiny philopodia when they are circulating in the blood. Okay, but when they are activated, the philopodia will increase in the size and number. Okay, and they also have random openings of channel system which is called as surface connected canalicular system. Now what is surface connected canalicular system? Let us see. So this is a structure of platelet. So here you can see this is the canalicular system and it is connected with the surface. Okay, or it is opening in the at the surface. So it is called a surface connected canalicular system. Now the function of this SCCS is it will increase the surface area of the platelet. Okay, because during most of the exocytosis the cell membrane will be utilized. Okay, or during, okay, so it will increase the surface area of the platelets and also it will act as a channel for exocytosis. That means these granules which are present in the platelets, so these are the alpha granules and dense granules, so they will open or they release their content into this canalicular system. Then this the material or the which is exocytose, it will enter into the plasma. Okay, so it will act as the conduit for exocytosis of granules. Okay, and it will also act as a reservoir for the plasma membrane. Okay, now we have surface connected canalicular system and we also have the closed system, dense tubular system. Okay, so this is dense tubular system, that means it is the closed system and it is nothing but the smooth endoplasmic reticulum which they obtain from the megakaryocyte. Now what is the function of smooth endoplasmic reticulum? So it will act as the because the dense tubular system, it will act as the reservoir for the calcium. Okay, calcium is very important for coagulation, so platelets have large reservoir for calcium. It is also required for exocytosis, okay, and also for the movement. You know, now you are learning the muscles. Okay, so calcium is required for actin myosin interaction and all these things. So calcium, so this dense tubular system acts as the reservoir for the calcium and also for the synthesis of prostaglandins. Okay, so these are the two canalicular systems, one is surface connected and one is dense tubular system. Now now we have two types of granules. Dense bodies, they are like bullseye, they have the bullseye appearance. Okay, so these are the dense body, and another type of granule is alpha granule. Okay, so these are the two main important types, and other granules are also present like lysosomes, okay, and peroxisomes, they also contain, they have their contents which are helpful for breakdown of products like catalase and lipase. Okay, now they have the glycogen granules also, which will act as a material for generating ATPs. Okay, they have the mitochondria. Then what else we have? We have the microtubules and microfilaments. So these microtubules which are present beneath the cell, plasma membrane, they will give the shape to the platelets, and they are also important in the for the movement of the platelets. And they also have the microfilaments. Okay, actin, myosin, thrombostatin, they are important for contraction of these platelets. Now the platelets will contract to remove the granular content as well as for the clot retraction. Now, okay, once there is formation of blood clot, the size of the clot will decrease after 1 hour. Okay, that process is called as clot retraction, and platelets play very important role in this clot retraction because they have actin, myosin, thrombostatin. So these are all contractile proteins. So with the help of these contractile proteins, they can contract or they can decrease in their size, and they will help in clot retraction. Okay, so this is about the structure of the platelets. Do you have any doubt in it? So even though these are only cell particles, they perform very important function. Okay, and they are, anybody wants to ask anything? Okay,
What is an alpha granule?
what?
Alpha granule.
हाँ, we will see about it. ठीक है, so हाँ, let us see one by one about platelet surface, membrane structures, cytoskeleton and granules. Okay, like the other cells have the plasma membrane, like that plasma platelets also have the plasma membrane, which is trilaminar. Okay, there are 3 layers, and its width is 20 nanometer. So what is the function of this plasma membrane? So they have various types of glycoproteins and lipids into their bilayer, and they act as the receptors for various things, like for binding with the endothelium. Okay, and they also provide receptors for binding of the clotting factors. Okay, so they are very important. So these glycoproteins and lipids, they are very important because they provide the permeability for various ions. Okay, they act as agonist for stimulation. Okay, and they are important for platelet adhesion, activation, secretion and aggregation. Now initially, when there is trauma to the blood vessel, the event which is taking place is formation of platelet plug. Okay, now for the formation of platelet plug, it is very important that the platelet should have the sticky surface. Okay, so that they will adhere with each other. Okay, and they will like this, they adhere with each other, they aggregate and they form the platelet plug. So this platelet surface is very important role which plays in the coagulation. It will provide the surface for attachment to the various clotting factors. Okay, once they attach to the platelets, the other factors will come and they activate that clotting factors, and the clotting mechanism will it will take place on the surface of these platelets. Okay, formation of fibrin, it will take place on the surface of platelets. So it is they will provide the platform for these clotting factors to perform their function. Now, so the surface, on the surface they have the platelets on the surface, they have the coat of glycocalyx. Okay, it is formed by the lipids, sugars and proteins. Now what is the importance of this glycocalyx? So whenever there is no injury to the tissue, okay, or when the endothelium is smooth, there is no any trauma to the endothelium, then the endothelium also has a layer of glycocalyx on its surface, and it is also smooth, and platelets also have the glycocalyx on the surface. So they will repel with each other. This sialic acid present on the platelet, it also will give the negative charge to the platelets. So they will repel each other, means endothelium and platelets will repel each other and they will not stick with each other, and there will not be any formation of the thrombus. Okay. Now if the endothelium becomes rough, okay, then what will happen? It will activate the platelets. So and it will lead to formation of the clot, okay, or the thrombus. So it is very important to avoid the formation of clot in the flowing blood. So it is very important to have the smooth surface which is covered with the glycocalyx, okay, so that the blood will remain in the fluid state, okay, otherwise there will be formation of blood clots. Now so in the plasma membrane we have seen that it has surface connected canalicular system. It also has microfilaments and microtubules, and it has mitochondria for the formation of ATPs, and dense tubular system of smooth endoplasmic reticulum, which is the main function of this smooth endoplasmic reticulum is storage of calcium, which is required for exocytosis, for contractility, okay, and also for coagulation. Okay. PL3s mean plasma proteins. Now, so there also means smooth endoplasmic reticulum is also required for the synthesis of prostaglandins, okay, which are also important for coagulation. So today we will see about the structure of platelets and its applied aspect, like what is the importance of calcium, prostaglandins, and all the contents of alpha granules, that we will discuss in the next lecture during hemostasis. Okay. So these are the granules which are present in the platelets, like dense bodies are there and alpha granules are there. So the dense bodies, because of their appearance under electron microscope, this name is given. Okay. Now the content of dense bodies is they have ATPs, ADPs, serotonin, calcium, prostaglandins, GDP and GTP. Okay. Now this ADP is very important content of platelets, okay, because it will help, it will cause activation of other platelets also. Once it is released from the dense bodies, it will activate other platelets also, and other activating function of other activating factors or stimulators, it will be increased by this ADP. Okay. So ADP is very important for activation of platelets. Now, so it is present in the dense bodies. There are around 4 or 5 dense bodies per platelet. Okay. Now we have another granules that is called as alpha granule. Now the contents of alpha granule are platelet factor 4, which we have already discussed. It is involved in platelet factor 4 and thromboglobulin. Both are involved in negative feedback regulation of thrombopoiesis. They inhibit platelet formation. Okay, that is negative feedback regulation. So alpha granules have platelet factor 4, thromboglobulin. They have adhesive glycoproteins like fibrinogen, von Willebrand factor. So von Willebrand factor is important because it helps in binding of platelets with the endothelium. Okay, and its deficiency will cause bleeding disorder. Now we also have fibronectin, thrombospondin, vitronectin in the in these alpha granules. Okay. Now we also have coagulation factors, factor 5 and 11. Okay. Now what is 5 and 11 we will discuss in the next lecture. So we have various coagulation factors up to 13. Okay, these are Roman numerals, they are given to the various coagulation factors, we will discuss in the next class. Now they have also mitogenic factor, platelet derived growth factor and transforming growth factor. So these are the growth factors which are involved in healing of the wounds. Okay, so this leads to growth of the fibroblast, okay, and angioblast. So they will help in the healing of the wounds. So they also have lysosomes, okay, and the contents are released. So the contents from the lysosomes are released very slowly, okay, and they have the elastase, collagenase and aryl sulfatase. So they cause breakdown of the tissue. you know that. So they are released very slowly and they require stronger agonists for release of these contents. And microperoxisomes are also there, which contains catalase. So these are all the contents of their granules. Now what are the conditions in which there is increase in the thrombocyte count? Okay, now the normal thrombocyte count is 1.5 to 4 lakhs per cubic liter of blood. Okay, so okay. Now what are the conditions in which thrombocyte count will increase? Now most of the platelets, they are sequestered into the spleen. Okay, so only 2/3 platelets which are present in our body, they are present in the circulation. Remaining 1/3 will be present in the spleen. So whenever there is any removal of the spleen or administration of epinephrine, so administration of epinephrine will lead to movement of these platelets from, they will remove the platelets from the spleen and then they will enter into the circulation. Okay, so this administration of epinephrine will increase the thrombocyte count by causing movement of these platelets out of the spleen. Now in splenectomy, obviously spleen is not there, so all the platelets which are developed, they are present in the circulation. After trauma, obviously there will be increase in, after trauma surgery there will be increase in the platelet count. Okay, and during stressful condition also there will be the platelets will enter into the circulation from the spleen and there will be increase in the platelet count. Now what are the causes for thrombocytopenia? That is very important. Okay, because mostly we come across the conditions which are leading to decreased platelet count, especially during rainy season. Okay, because we have the many patients having dengue, or during any some viral conditions, viral fever also, there will be decrease in the platelet count. Now if the normal count is between 1.5 to 4, okay, lakhs per cubic millimeter. So if the platelet count is greater than 1 but less than 1.5, okay, what will happen? So there will not be any symptom. Okay, bleeding time will also be normal. Now what is bleeding time? Bleeding time means it is the time required to stop the bleeding. Okay, so in our lab we will perform the test. So you have to prick your finger with the help of a needle. Okay, and then you have to observe the time required to stop the bleeding. So that is the bleeding time. Okay, so the bleeding time, it is mostly depends upon the function of the platelets. So if the platelet count is less, the bleeding time will become more. Okay, so if the platelet count is more than 1 lakh per cubic millimeter, then the patient will not have any symptom and the bleeding time will also be normal. If the platelet count is between 50,000 to 1 lakh per cubic millimeter, so if there is severe trauma, then there will be more bleeding, and bleeding time is only mildly prolonged. Okay, there is not much increase in the bleeding time. Now if the platelet count is less than 50,000, okay, then we can observe this bruising under the skin or purpura. Okay, so after minor trauma, okay, so there are different tests which are used to know whether the platelet count is less, or even though if the platelet count is less, it will not sometime manifest. So there are different tests to increase the pressure over the skin, like by applying BP apparatus. You all know the sphygmomanometer. So sphygmomanometer cuff can be applied and pressure can be increased to increase the and then we see whether purpura will appear in the skin or not. Okay, so there are different tests which can be used. So if the platelet count is less than 50,000, after minor trauma there can be appearance of purpura, or there can be bleeding from the mucous membrane after surgery. Now if the platelet count is less than 20,000, there are chances of spontaneous bleeding. Okay, and there are also chances of having internal bleedings. So it is observed in dengue fever. Okay, so what we do, if the platelet count is less than 20,000, then we give either fresh blood, because if the blood is stored for longer period, then the platelet count will decrease in that blood. So either fresh blood or platelet concentrate will be given to the patient. Okay, now what are the causes for thrombocytopenia? So you all know that platelets develop into the bone marrow. So either bone marrow infiltration by the tumor, or if there is fibrosis of the bone marrow, or if there is a plastic bone marrow, then there will be decreased platelet count. Okay, now splenic sequestration of platelets. So normally 1/3 platelets are present in the spleen. So if there is spleen enlargement like by tumor, okay, or by splenic congestion, then there will be increased sequestration of these platelets into the spleen. It will also decrease the platelet count. Now there can be increased, either there is decreased production or increased destruction of platelets. Okay, now there for the destruction of platelets, there are two mechanisms, either non-immune or immune mechanisms. Now in non-immune, there will, when there is presence of any vascular processes like stent. Okay, stent we use for whenever there is any blockage in the coronaries. Okay, then we use the stents. So in that case, the platelets will attach to that surface, and then there will be breakdown of platelets. So this is non-immune destruction of platelets. Also in sepsis and disseminated intravascular coagulation, like whenever there is any severe bacterial infection, okay, it will also stimulate the coagulation, and in that process there will be increased destruction of platelets. Okay, now immune destruction will be there if there is autoantibody formation against the platelets, or because of the drug hypersensitivity. And in viral infection, one of the mechanism is formation of antibodies, and they will destroy the platelets. So in dengue fever, there are so many mechanisms which lead to breakdown of platelets, like activation of antibodies, then secretion of cytokines. Okay, so there are multiple mechanisms, complex mechanisms are involved in breakdown of platelets during dengue fever. So in other viral infections also we observe thrombocytopenia.
Ma'am, what is the meaning of purpura?
Purpura means these are small purple patches which are present beneath the skin because of the capillary leaks. Okay, because normally what happens, whenever there is any capillary leak, it will be occluded by the platelet plug. So if the platelet count decreases, whenever there is some capillary leaks, there are thousands of capillary leaks developed during day-to-day activities. So they will not be plugged by the platelets. So we observe purplish patches. So if you have dengue, मतलब अगर फैमिली में किसी को हुआ है, या फिर इफ़ यू कैन सी इन द मतलब ऑनलाइन भी देख सकते हो डेंगू के पेशेंट्स में. सो इन दिस पेशेंट्स वी ऑब्जर्व द पर्पल colored patches over the skin. ठीक है, that is called as purpura.
Vascular prosthesis.
That means stents. हाँ, vascular prosthesis means stents we apply ना. Whenever if the patient has myocardial infarction, okay, if there is blockage in the coronaries, then we use the stents to inflate the arteries. Okay, so these are the foreign surface of foreign bodies. Okay, so the platelets can adhere to that stent, and then there will be destruction of the platelets, because the platelets have the tendency to adhere to the surface. Whenever they come in contact with the any surface, foreign surface, they will adhere to that surface and they will get activated. Okay, so there will be increased destruction of platelets because of these vascular processes.
Thank you, ma'am.
Okay, so now, so this primary hemostasis and secondary hemostasis we will discuss in the next class. So in the primary hemostasis, there will be formation of platelet plug which occurs in seconds, and which is important for stopping of bleeding from capillaries, small arteries and venules. And secondary hemostasis is the formation of blood clot. Okay, so it involves the clotting factors. There are various clotting factors, and it is very important for larger blood vessels. Okay, so platelets play very important role in primary hemostasis. So whenever there is small injury to the blood vessel, the main mechanism for stoppage of the bleeding is formation of platelet plug. Now that mechanism also we will discuss in the next class. Any difficulty related to this class?
What is sepsis?
Sepsis means it is the whenever there is systemic bacterial infection, okay, that is called as sepsis. So it is a serious condition. Now in the last class there were few questions. So what is the role of interferons in the immunity? Okay, there was some confusion. That is what is the how these interferons act. So they form protective ring of uninfected cells and inhibit protein synthesis. Okay, so what is that? So this is the mechanism of action of interferons. Okay, so the interferons is that a non-specific proteins. So these are released into the circulation. They are mostly involved in viral infections. Okay, so the interferons are important to recover from the viral infections, and they themselves don't have any mechanism to destroy the virus. Okay, but they stimulate or they lead to formation of other proteins which will lead to breakdown of this virus. Okay, now how they will act? So suppose this is the virus which infect the cells, okay, like leukocytes. So once these so these viruses, what they do, they will enter the cell. Okay, that is the normal property of the virus. These are intracellular organisms. So they enter the cell, and inside the cell they will proliferate. Okay, now once they enter inside the cell, they will lead to the cells or the leukocytes, they will lead to formation of these interferons. Okay, now these interferons, once they are produced, they will act on the neighboring cell. Now you can see here that this cell is not the virus is not present inside the cell. Okay, so it is a non-infected cell. So on this cell, these interferons will act, and they will start the expression of various proteins. Okay, that is called as interferon stimulated genes. So they activate these interferon stimulated genes, and they will start producing the proteins from this in this cell, and this is called as priming of this cell. Okay, now if the virus attaches with the cell, or if the virus enters inside the cell, then these proteins which are secreted by the gene expression, they will cause breakdown of this viral mRNA, and they will prevent the viral protein synthesis and growth of the virus. Okay, now like this, and also they will cause MHC1 expression, so that it will stimulate the cytotoxic means T cells. Okay, and they will cause breakdown of this cell, and there will be ultimately the growth of the virus will be prevented, and the patient will recover from the viral infection. So this is a mechanism by which these interferon are activated in the viral infection, and they will protect us. They protect us from the different viruses. Okay, they themselves don't have any function. They will activate only the expression. They will lead to expression of the genes which are involved in the formation of various other proteins. Okay, like protein kinases and other enzymes. Now there was another question, that is RBCs, whether RBCs have any HLA antigen or not. So normally we say that HLA type 1 is present only on all nucleated cell and on the platelets. Okay, but the HLA antigen is only weakly expressed by the RBCs, and means in the textbooks it is not given, but in the articles it is given that around 50% of the French population has HLA type 1 antigen on the RBC. But for the all your Viva questions and all these questions, HLA type 1 antigen is expressed only by the nucleated cells and by the platelets. Okay, now why and how they are protected from the our immunity? Okay, because during pre-processing of WBC, T cells and B cells, they are exposed to the various self antigen, various self peptides by with the help of this HLA type 1 and type 2. So HLA type 1 and type 2 is required for the expression of these self antigens to the cytotoxic cells and to the helper cells. Okay, so even though RBCs don't have HLA type 1 antigen, it will not lead to anything. Okay, because the self antigens which are present on the surface of RBC, they will be expressed to the T cells as well as B cells during their pre-processing, and the cells which are not reacting to the self antigen, they only will be released into the circulation. So there will not be any auto immunity against our RBCs. Okay. So what happens in dengue? So there are multiple mechanisms for formation of auto antibodies against formation of antibodies, because on the surface of the destroyed platelets we observe the antibodies. So antibodies are also involved, cytokines are also involved. So there are multiple mechanisms which lead to breakdown of platelets. So there will be decrease in the platelet count in the dengue fever. Okay, so tomorrow we will discuss about hemostasis. There will be 2 lectures, one on hemostasis and another one is on blood group. So please start reading the blood. Okay, now what happens in COVID? Is there any increased thrombosis in COVID-19? Have you heard about it? Are you reading now the pathophysiology of COVID-19? Because it is very relevant to hematology lectures. Okay, so that we will discuss in the next lecture. Okay. Okay, now then bye bye. Dr. Dinu will take next class.
Students, good morning all of you. So we shall start with the nerve muscle class. Am I audible to everyone?
Yes. Yes.
Yeah, and
Yes, sir.
the screen is also visible, right? Slide is also visible. Right. Okay. I just want to give few announcements that I had shared with you. एक तो lecture आप लोगों ने देखा होगा. Most of you I think have gone through, and this PBL session. That PBL is problem based learning. You might have already been exposed to it. What we will be conducting is a slightly modified version of PBL, which is called case based learning. There are some differences between the two. So Saturday afternoon 2 hours we would be having this. You will be basically working in small groups. स्मॉल ग्रुप्स मतलब 10 लोगों के ग्रुप्स में डिवाइड करेंगे आप लोगों को. ऑनलाइन ही होगा तो ऐसा होगा. तो आई हैव शेयर्ड फ्यू मटेरियल ऑलरेडी विथ यू. कुड यू आई होप यू हैव रिसीव्ड इट, राइट? वो जो CBL का मटेरियल मिल गया. हाँ, ईमेल में ही भेजा था, सेम ईमेल में. या so please go through that. So that is those are the preparatory resources. So the basic difference between CBL and PBL is that PBL में it's like explorative learning. So you only explore, you read the case or problem and you identify कि आपको क्या जानना मतलब required है, what do you want to what do you look for to understand the case or whatever, and then you start searching yourself. And that's the way. It's more explorative in nature. But CBL में we give the preparatory material. तो एक material है वो lecture ही है न्यूरोमस्कुलर ट्रांसमिशन का, इसलिए दैट इज़ व्हाई आई गिव इट एज अ वीडियो लेक्चर, बिकॉज सो दैट यू कैन सी इट मल्टीपल टाइम्स इफ यू वांट टू, एंड एट योर कन्वीनियंस एज वेल. और जो मटेरियल जो मैंने ईमेल में भेजा है वो क्लिनिकल प्रॉब्लम सॉल्विंग के लिए इंपॉर्टेंट है. सो व्हेन यू लुक एट द सो बेसिकली वी विल हैव ए क्लिनिकल केस, एंड that case is actually a published case report only. मतलब it's not a fabricated case, it's an actual case. And so to understand the terminologies of that case, तो CBL में क्या होता है कि हमने जो मटेरियल पहले दिया वो पढ़कर आपका अवेयरनेस और नॉलेज बिल्डअप करके आएंगे. So that when you are working on the case, you are primarily looking at the case and analyzing it. Obviously you can look for relevant information online, offline, टेक्स्ट बुक जब आप लोग ग्रुप एक्टिविटी करेंगे उसके साथ साथ भी देख सकते हैं. बट टू बेस्ट मेक द बेस्ट यूज ऑफ द टाइम, it is advisable to get prepared so that at least you would know where you have the relevant information about that part of the case. तो जो मटेरियल मैंने भेजा है एक तो अप्रोच टू न्यूरोमस्कुलर डिसऑर्डर्स के ऊपर है, एक तो एक क्विजलेट है which is about the terminologies which are used to familiarize with the terminologies. फिर approach to neuromuscular disorders, and the conduction velocity, nerve conduction velocity is a practical for you, which will be teaching when the laboratory exercises start. तो उसके बेसिस उसका कैलकुलेशन कैसा होता है, इंटरप्रिटेशन कैसा होता है, उसका भी एक मटेरियल मैंने भेजा है. इट्स नॉट नेसेसरी दैट यू विल अंडरस्टैंड एवरीथिंग व्हिच इज रिटन, बिकॉज इट्स मोर क्लिनिकल ओरिएंटेड एंड यू आर जस्ट अभी शुरू हुआ है तो उतना समझ नहीं आएगा. दैट्स नॉट ए प्रॉब्लम, वी कैन डिस्कस इट लेटर एज वेल. एंड बट बी prepared with the material which we have shared to whatever extent possible, and it will be a good experience, I can assure you, because you get to see a clinical case with the history, with the examination, physical examination findings, and the investigations done, laboratory tests and other things done, and how do you reach to the diagnosis. It's not the diagnosis which is important here, but how do the normal physiology get disturbed. कि एक डिसीज़ कंडीशन होता है तो उसका एक मैकेनिज्म होता है, एंड देयर इज़ अ रीज़न बिहाइंड इट. तो उसकी वजह से how does it affect the normal physiology, and how would it result in different clinical manifestations. So when you are learning physiology, you are studying the normal functioning of the body, but at the same time you should also be able to correlate or relate कि अगर कोई डिसीज़ कंडीशन होता है और नॉर्मल फंक्शनिंग को it is getting affected, then how does that change in normal physiology manifest as the clinical features or the clinical manifestations or the symptoms. तो वो mechanisms हमको समझना है. So that is what will be the core element of that case based learning. So be prepared and participate actively and interact actively to learn about it. ठीक है. So we move on to today's class. In any specific queries on neuromuscular transmission, we can take it up later or during the case based learning session itself. So today we are moving on to muscle. Now muscle, as you know, we started with cell membrane, membrane electrophysiology, फिर resting membrane potential के बारे में बात किया कि how is it generated. उसके बाद electrical signaling के बारे में बात किया कि action potential is the signal and how is it generated. कि जब आप electrical excitation देते हैं तो how is action potential generated and propagated. And then we talked about कि because we are talking about nerve muscle physiology, we need to tell कि we need to understand कि nerve से जो signal आता है वो muscle के how does it excite the muscle, and there is a gap between the two. How does it cross? So that is what was the neuromuscular transmission lecture all about. उसके बाद now we are coming to the muscle. Now muscles, as you know, they are actually kind of biological machines. Now they can convert chemical energy into mechanical work. So basically they are biological machines, and they are almost 40 to 50% of the total body mass. And it could be variable, but 40 to 50 is the range normally seen. So muscle, there are different muscles. Muscle types के बारे में अगर बात करते हैं तो skeletal muscle is what we are going to talk about in our lecture today. So muscles, as I said, they can do mechanical work. So they can shorten, they can build up tension by contracting. So they are present in not only as skeletal muscles. Skeletal muscles are the muscles which will move the skeleton. So skeletal muscles have a point of origin, point of insertion, और जब वो shorten करते हैं या contract करते हैं तो it will move a particular joint or a limb. So that is how it works. So they are again doing mechanical work using the chemical energy. Cardiac muscle, the heart wall is made up of cardiac muscle, which also does work again by contracting and relaxing regularly in a rhythmic manner. Contracting matlab it will generate pressures, wall tension and pressure to push the blood out of the cavity of the heart, and then relax and receive more blood from the venous side, again push, contract, eject it. So this is how they work. And you also have smooth muscles जो आपकी visceral organs की wall पे है, जैसे bladder. When you are emptying the bladder, again you need to generate force to push the content outside. When similarly intestinal smooth muscles. So you have different types of muscles. So here you see the histological pictures. So you can see the skeletal muscle and the cardiac muscle and the smooth muscle here. Now skeletal muscle and cardiac muscles are known as striated muscles. Why are they called striated muscles? Anybody who can say some? Yeah, please.
Continues the differential arrangement of actin and myosin filaments in a specific manner to form a pattern.
Yeah, so striations are that vertical lines you can see in this picture. ये हिस्टोलॉजिकल पिक्चर में आपको एक मतलब लाइट एंड डार्क स्टेन striations मिल जाता है. So exactly as you said, and that is because of the repeating arrangement of thick and thin filaments in a very well aligned manner. We will be coming to that in the subsequent slides. So skeletal muscle or cardiac muscle में you find these striations because of their similarity in the arrangement of thick and thin filaments in the form of myofibrils, and so that is why we have that striations. Whereas smooth muscle, you don't have that very repeating and well organized aligned arrangement. So in smooth muscle you don't get the striations. So this is basically a structural difference. Obviously there are a lot of functional differences also, and today we are starting with skeletal muscle. जब आप लोगों का कार्डियोवास्कुलर सिस्टम फिजियोलॉजी होता है तब कार्डियक मसल के बारे में आप लोग डिटेल में पढ़ेंगे. स्मूथ मसल आई विल ओनली बी कवरिंग एज द इंट्रोडक्टरी पार्ट. दैट विल बी द लास्ट लेक्चर्स ऑफ दिस सीरीज स्मूथ मसल के ऊपर. सो उस टाइम हम दोबारा उसके कंपेयर एंड कॉन्ट्रास्ट करेंगे कि स्ट्रक्चरली और फंक्शनली हाउ स्मूथ मसल्स आर डिफरेंट फ्रॉम स्केलेटल मसल इन देयर स्ट्रक्चर एंड फंक्शंस. सो वी विल बी रिविजिटिंग दैट. नाउ कमिंग टू द ग्रॉस स्ट्रक्चर, तो ये एक्चुअली एक पिक्चर है जिसमें यू कैन सी द बोन and there will be fascia and there will be tendon, basically the tendinous origin of the muscle, and there will be deep fascia associated with that, And when you see, there is an outermost covering which is called the epimysium. Within that you have the skeletal muscle, and in the cut section, as you can see here, you would see that the muscle fibers, muscle fiber matlab muscle cell, group of muscle fibers or muscle cells are they are together in the form of fascicles, which are so these are all fascicles. So ये एक fascicle है, एक दूसरा fascicle है. तो एपीमाइसियम का बाहर का कवरिंग एपीमाइसियम का होता है, उसके अंदर फिर each fascicle is surrounded by a perimysium. Okay, perimysium. Or when you take that fascicle out, then you have the individual muscle cells, and each muscle cell के अंदर भी in the cut section you see cylindrical structures. Now these cylindrical structures are called myofibrils. We will come to the detailed ultrastructure of that in the subsequent slide. So epimysium, muscle fascicles separated by perimysium, and in the perimysium you will find the arteriovenous and the neurovascular bundle. मतलब आर्टरी, वेन और नर्व जो इनर्वेट करते हैं और आर्टीरियल सप्लाई और वीनस आउटफ्लो होता है, वो सब इस फेशियल लाइनिंग के अंदर होता है. सो यू कैन सी दिस हियर, एंड so this is how the structure is actually when you see it in the cut section. So there are many myocytes which are held together in the form of fascicles. Neurovascular supply to muscles course through the connective tissue sheets epimysium, perimysium, endomysium तक आकर वो इनर्वेट करते हैं, arterial blood supply और venous outflow आता है. And the tendons, they are the bundles of connective tissue rich in collagen known as tendons, attach muscles to the bone, and this attachment is very essential for transmitting the force. मतलब these are like elastic elements which are attached to the bone at both the sides, and when muscle contracts it pulls from both the sides, and that's how the joint moves. So that's what we will be seeing further. And now why are the muscle cells called muscle fibers? Because muscle fibers are long. These are individual cells, but because of their shape is cylindrical, most of them, and they are long. They could be as long as 25 centimeters in some of the muscles, though their diameters would be 10 to 100 micrometers. So they are very slender, thin, long fibers. So that is why muscle cells are called muscle fibers. And you would also see that the nucleus of these cells, they are multinucleated cells, and the nucleus is generally pushed to the sides. This is the nucleus actually. It's pushed to the side because the cell is filled with the myofilaments arranged in the form of myofibrils. So that is why you have the nucleus displaced to the side. If you see any particular cell in the body, the cells have their own adaptations or their own way of differentiating into a form in which their functions are best performed. So as far as a muscle cell is considered, उसको basically क्या है कि force generate करना है. जब भी stimulus आता है, force generate करना है. उसके लिए thick filament और thin filament के बीच में interactions होना चाहिए. So the entire cell is filled with thick and thin filaments arranged in the form of sarcomeres and myofibrils. We will be coming to those structures subsequently. और बाकी सारे structures जो है energy देने के लिए mitochondria चाहिए, because oxidative phosphorylation उसके अंदर होता है. So mitochondria will be there, endoplasmic reticulum will be there, but majorly the cell is filled with these myofibrils, because that is what they are capable of generating power, or they are capable of doing work, mechanical work when there is a stimulus. So again, further zooming into what we have already seen. So again, whole muscle है ये, उसके अंदर fascicles है, fascicles के अंदर you have the cylindrical cells which are muscle fibers, which are called muscle fibers. You can see the blood supply going. You can see the striations here. Now these striations is because of the, now this is a single muscle fiber, एक cell है, उसका cut section अगर देखोगे तो उसके अंदर it is studded with lot of cylindrical structures again, and these are made up of thick and thin filaments, and one of this cylindrical structure which is in a linear way, you can see the thick filament, thin filament, thick filament, ऐसे करके जो repeating arrangement होता है in a linear fashion, that's called a myofibril. And the myofibril is composed of myofilaments of two types majorly, thick filament and the thin filament. So this is the thick filament, which is majorly made up of myosin, and the thin filament, which is majorly made up of actin filament as actin. So these terminologies are very important, students. कि मसल फाइबर क्या होता है? फाइबर इज अ सेल. मायोफिब्रिल क्या होता है? मायोफिब्रिल इज रिपीटिंग अरेंजमेंट ऑफ थिक एंड थिन फिलामेंट्स, and every muscle fiber is filled with this repeating arrangement of myofibrils in a well aligned manner. मतलब अलाइनमेंट ये है कि जहाँ पर इसका थिक फिलामेंट है, नाइबरिंग मायोफिब्रिल का भी थिक फिलामेंट ऑन यही होगा. यहाँ पर थिन फिलामेंट्स ऑन है तो नाइबरिंग मायोफिब्रिल्स का भी थिन फिलामेंट्स ऑन यही होगा. So that is how they are made, and within the myofibrils you have the filaments, myofilaments, and these are the thick filaments and the thin filaments. So repeating pattern of arrangement of thick and thin filaments within the myofibril along with the highly organized alignment of adjacent myofibrils lead to the striated appearance. तो striated appearance की रीज़न ये है, एक तो थिक थिन करके अरेंजमेंट है और एडजेसेंट मायोफाइब्रल्स का अलाइनमेंट एकदम मतलब परफेक्ट है कि जहाँ पर इसका थिक है तो इसका भी नाइबर्स का भी थिक फिलामेंट जोन वहीं पर आता है. सो व्हेन यू सी अंडर द माइक्रोस्कोप यू विल हैव दिस जोन्स रिपीटिंग, एंड दैट इज व्हाट यू सी एज द वर्टिकल striations which you would observe in the muscle fibers. So now we look into what you call as, and I'm sure these terminologies names are familiar to you even in your 11th 12th classes की ये सब होता है in a less detailed manner कि Z disc, M line, exon, I band, ये सब I am sure it will be familiar to you, but we'll be going into more details of it. So we know that there is a repeating arrangement of thick and thin filaments. Now if you take two Z discs, मतलब Z discs are the places where the thin filaments are attached. So thin filaments will be projecting in opposite directions from the Z disc. इस तरफ भी एक thin filament होंगे, and from here also इस तरफ also you have the thin filaments. And at the central part you have an M disc or an M line जहाँ पर thick filaments are incorporated, and thick filaments also project to either side of this M disc. So Z disc से thin filaments project होता है, M disc से thick filaments project होता है. Now you have this I band and A band. Can anybody tell me how did this name A band and I band emerge? कैसे origin हुआ इसका term? Who can speak?
Isotropic and anisotropic.
Yeah, what does that mean, anisotropic and isotropic?
Light can pass through and light can't pass through.
Yeah, please one at a time. Yeah, so बताइए. Who was speaking now?
Sorry, on pass.
Yeah, please introduce your name and tell me so that I know who is speaking. Yeah. हाँ.
Pratik Agarwal.
Okay, बताइए.
Sir, on passing polarized light, the isotropic bands appear light in color, whereas anisotropic appear dark in color.
Why is it so? Why is it dark? Anisotropic क्यों dark होता है?
The isotropic, iso means one or the same. So in I band there is only one type of filament, and in A band there are two types of filaments.
Okay. So why would that lead to dark and light? That is the question. So isotropic. बताओ please continue.
Because of crowding of proteins.
Yeah. So refractive indices are different. So refractive indices different होगा तो refractive indices different होगा तो
Maybe because electron density is more.
no, but this is not an electron micrographic picture. This is polarizing light microscopic picture जिसमें आइसोट्रॉपिक और अनआइसोट्रॉपिक. एक्चुअली क्या है ना, इलेक्ट्रॉन माइक्रोस्कोपी बाद में आया है. जब ये डिस्कवरी चल रहा था तब इलेक्ट्रॉन माइक्रोस्कोपी अवेलेबल नहीं था. इलेक्ट्रॉन माइक्रोस्कोपी के अंदर भी इलेक्ट्रॉन डेंसिटी के हिसाब से देखते हैं. बट व्हेन दिस टर्मिनोलॉजीज वेर गिवन it was based on the light microscopy and this isotropic and anisotropic. Soeb कौन है? He has posted a explanation based on refractive indices. Can you expand further on it?
Sir, when the refractive indices will be different, then light will behave differently, and when the both, actin and myosin will be there, and the refractive indices are different, so light will behave differently.
So can you just, so I will request you to go to little detail of this. Uh, uh, Birefringent and anisotropic. It's more than birefringence. You can look into this. When the refractive indices are different in different axes, then when you send the light, there is a chances of destructive interferences there, which does not happen when it is in an isotropic band, because the light waves will be delayed because of the deceleration happening, because the refractive indices are different. So there would be chances of destructive interference, which would make the A band look dark. Obviously the reason is because you have two different types of filaments and proteins there, and so you can basically look into little more details. Basically I would like to say the optics or the physics of it, because you are the right people to do it. तो वो थोड़ा और डिटेल में देखकर नेक्स्ट क्लास में मुझे बता देना. Whoever interested can go through and come up with a much more clearer picture कि why is it darker. Obviously the things are around anisotropic in nature of the refractive indices, and isotropic matlab refractive index is homogeneous or same in that particular zone. So we can discuss it up later, but look into that. Yeah, I'm getting more replies on this, so we'll take it up in the next class. Get more clarity on why it is dark. The question is, when the light is shown, why is A band becoming dark and why the I band is light? How could we explain that on the basis of refractive indices and what is happening there? Yeah, so if you come to the molecular arrangement, you can see that, as we said, so this is from based on this light microscopic images, and you have this M line here, like you have the M line here, and the basically the overlapping zone is the A band starts at one side and ends on the other side, where you have the overlapping. So the thin filaments are here. The I band is the place where you don't have the overlap. So I band is on either side of the Z disc where you don't have the overlap, and A band is basically the area where you have overlap, but then there is also a middle portion which is just adjacent to the M line where you don't have the overlap. This is again a thick filament alone zone, and that is called the H zone, and obviously it will look lighter because of the same reasons ki isotropic and isotropic nature ki. So and these short forms of VZ, AM वगैरह जर्मन वर्ड से, जर्मन लैंग्वेज की टर्मिनोलॉजी से ओरिजिनेट किया हुआ है. जो ओरिजिनल रिसर्चर्स उस टाइम काम कर रहे थे इसके ऊपर, उन्होंने इसी वर्ड्स के बेसिस पर ये सब शॉर्ट फॉर्म्स दिया हुआ है. सो जस्ट फॉर योर अवेयरनेस आई पुट दिस वर्ड्स. सो मीनिंग ऑल्सो लाइक Z disc basically mean interval discs in German, Zwischenscheiben, and divide each myofibril into a linear series of repeating contractile units. So what you call as sarcomere is between two Z discs, because that would comprise of I band, half of the I band of this side, half of the I band of this side, but basically thin filament यहाँ से, thin filament यहाँ से, thick filament center पे. So when thick filament interact with thin filament, this sarcomere is going to shorten. That's what you understand. So sarcomeres are actually the contractile units कि जिसमें thick and thin filament की interactions होते हैं. So sarcomere is a structural and functional unit, and it is bound by two consecutive Z discs or lines, and roughly 2.2 microns, 2 to 2.2 microns की length होता है in the resting muscle. And as I said, thick filaments are bound together here, so there would be some a lattice here. जब हमने when you see these images, these are represented as lines, but don't consider these as lines. Imagine that these are three-dimensional structures. So perpendicular to the screen where you are looking at, एक disc है. So it is an M disc, it is an Z disc, and there is a lattice here जिसमें और भी proteins होते हैं, which will be able to hold it up. So this is how it is going to be working, and so thick filaments are bound together at the center of the sarcomere by the M line or M discs, which is name again meaning middle of the discs, and thin filaments are inserted into Z discs, and their free ends overlap with thick filaments, which we already know. A band is anisotropic, which is the darker overlapping zone. I band is the lighter non-overlapping zone, which is thin filament zone basically. Okay. So H zone is a lighter non-overlapping zone on either side of the M line. So this I had already told you about कि there is a portion जहाँ पर overlap नहीं है, सिर्फ thick filament की जो the body of the molecule, these are the cross bridges, the head part, head, neck and tail होता है. तो जहाँ पर tails आ जाता है वहाँ पर आप लोग overlap नहीं देखेंगे. We'll go into the detailed structure of this molecule subsequent slides में. So that is the place where you have again H zone. Now when you stretch the muscle, what will happen to I band and what will happen to H zone? Can anybody tell? When you stretch the muscle, not when the muscle is contracting. तो stretch करेंगे तो क्या होगा? Dimensions of H zone and I band, that will increase and the overlap would reduce, so A band का overlapping area कम हो जाएगा और H zone और I band की dimensions बढ़ जाएगा. So this is just to explain that. This is again a schematic picture, वही है जो आप लोगों ने अभी देखा था. It shows more number of proteins and what I was talking about कि Z line बोलते हैं हम, लेकिन Z disc है वो, उसके अंदर और भी proteins होता है like desmin, plectrin. तो वहाँ से फिर आप लोग देखेंगे कि see the thin filaments, these bluish colored ones are thin filaments, and you can see a protein called nebulin which is wound over the thin filament. A thin filament is basically made up of filamentous actin molecules से बनते हैं वो. और ये एक important चीज़ है कि M disc proteins भी होता है, जैसे myomycin, it is also a disc. इसके अंदर बहुत सारे proteins हैं जो thick filaments को वहाँ पर hold करके रखते हैं, and so that there is a lattice arrangement and stability is given to the thick filaments. Now one important, very important molecule which is there is titin. Now titin का basically ये है कि it runs from the M discs on either side, it reaches till the Z discs, and it is closely associated with a thick filament, and it is an elastic protein. This is perhaps the largest protein which is there in our body. Its molecular weight is close to 3000 kilodaltons. So a big molecule, and it has something like elastic in the sense कि यहाँ पर folded जो structures दिख रहा है, जब muscle को अगर आप stretch करोगे तो this part will get stretched and that would build up force. So when a muscle is being stretched, it generates what you call as the passive tension. Passive tension बोलते हैं उसको. वो passive tension इसलिए build up होता है क्योंकि this elastic in the sense anything when the dimensions or shape is changed, if it generates force, that's called an elastic structure. So titin is an important molecule. It also plays a role in the keeping proper alignment of thick filaments. इसमें भी इसका role है, लेकिन it's also mechanically active. So we will come to that in some other classes as well कि titin का role क्या है. So you have many proteins. So this slide is not a very essential information नहीं है, ये आपके अंडरस्टैंडिंग के लिए रखा हुआ है, लेकिन बट व्हाट यू आर सपोज टू नो इज एट लीस्ट दिस मच कि थिक एंड थिन फिलामेंट, एम लाइन, इजेड डिस्क, ये सब तो पता होना चाहिए. ओके, नाउ जस्ट फॉर योर अंडरस्टैंडिंग दिस इज शोइंग अगेन द कट क्रॉस सेक्शंस. नाउ लेट्स सी व्हाट इज देयर इन इजेड डिस्क. तो इजेड डिस्क में व्हाट यू सी एज द green here, what is that? These are actually titin. Okay, you can see in the next cut section also. So as you can see in the slide, titin reaches till here. Z disc में titin का presence है, and titin corresponds with the thick filament. Thick filament doesn't reach till Z disc. We know about that. Thick filament stops at this extent. ये जो A band जहाँ पर शुरू होता है, लेकिन each thick filament is associated with a titin molecule which goes and reaches up to the Z disc. तो इफ़ यू सी थिक फिलामेंट जहाँ पर था वहाँ पर अभी टाइटिन मॉलिक्यूल्स मिलेगा, एंड दिस रेड स्पॉट्स आर द थिन फिलामेंट्स, एंड यू कैन सी द काइंड ऑफ क्रिस्टलाइन लैटिस काइंड ऑफ अरेंजमेंट इन द डिस्क विद द हेल्प ऑफ अदर प्रोटीन्स लाइक डेस्मिन, प्लेक्ट्रिन जो भी है बोल रहे थे वो सब यहाँ पर होगा इजेड डिस्क प्रोटीन्स. नाउ व्हेन यू जस्ट गो एडजेसेंट टू द इजेड डिस्क से हम इधर तक आ गए, तो यू हैव द आई बैंड जहाँ पर आपको ओवरलैप नहीं मिलेगा. जहाँ पर ओवरलैप नहीं है तो आप लैटिस काइंड ऑफ स्ट्रक्चर, मतलब वो नेटवर्क नहीं मिलेगा क्योंकि यहाँ पर आप इजेड डिस्क प्रोटीन्स नहीं मिलेगा आपको, बट यू हैव द थिन फिलामेंट्स इन अ हेक्सागोनल अरेंजमेंट विथ अगेन टाइटिन. Why don't you get thick filament here? Because there is no overlap here. So you will only see titin molecule which is crossing from the M line to the Z line. Again, that will be surrounded by thin filaments here, but you don't get thick filaments here. Now, when you reach let's go to the A band now. Let's go to from I band, let's go to the A band. Now, A band is the overlapping area. तो A band में आपको मिलेंगे थिन फिलामेंट ऑब्वियसली होगा और थिक फिलामेंट भी मिलेगा एंड इस थिक फिलामेंट के साथ यू वुड हैव दी titin also, though titin is not separately shown here. Thick filament and titin together hoga. So these two I band and A band arrangements are similar, except that in the I band you won't get thick filament or myosin basically, whereas in the A band you would get thick filaments. Uske baad if you come to, let's come to the M line. M line में क्या है? It is similar to Z disc, क्योंकि Z discs and M discs are the points where you have the lattice stability being established with the help of a network of proteins which connect all these structures and strengthens it and maintains it. तो वहाँ पर आपको वो मायोमेसिन जैसे प्रोटीन्स मिलेंगे, थिक फिलामेंट्स होगा, टाइटिन होगा, वो एक क्रिस्टलाइन लैटिस जैसे दिखेगा. फिर एज जोन इज द प्लेस वेयर यू डोंट हैव थिन फिलामेंट्स. ऑब्वियसली इन द एम डिस्क ऑल्सो यू डोंट हैव थिन फिलामेंट्स एंड एज जोन में भी आपको थिन फिलामेंट्स नहीं मिलेंगे. तो बेसिकली थिक फिलामेंट्स और एसोसिएटेड टाइटिन होगा यहाँ पर, और this is the structure. So this cut sections up carefully, maybe you are maybe little confused now because you have to carefully look through it. So what you need to understand is the similarity in the structure of M disc and Z disc, because there are other proteins which are stabilizing. That I band and A band, what is the difference? And understanding that titin is one molecule which is present throughout the system from M disc to Z disc. So यही है lattice structure of sarcomere. You can go through this. And now coming to the ultrastructure. Ultrastructure मतलब how these thick and thin filaments are individually, मतलब उसका what all molecules are going to make up this thick and thin filaments. So if you look at thick filament, it's made up of myosin molecules which are, suppose this is the M disc, you have the myosin molecules arranged in a bipolar manner. Bipolar manner की मतलब ये होता है, अगर यहाँ पर M disc है तो इस तरफ के molecules की head will be towards right and इस तरफ के molecules की head, this is the cross bridge head, will be towards the left. So you have a kind of bipolar arrangement on either side of the sides of the M disc. And if you look at the thin filaments which are emerging from the Z discs, you can see the double helical structure, just like your DNA molecule. You have the double helical structure, and this is made up of filamentous these individual globules or the globe-like structures are the actin molecules. जब actin, globular actin बोलते हैं उसको, globular actin polymerize करते हैं, it makes filamentous actin, so it becomes a filament. And on that you will find two other molecules which are very important. This is troponin and this is tropomyosin. So actin, filamentous actin as a double helical structure plus tropomyosin plus troponin would form the thin filament. Obviously nebulin is also associated here. उतना डिटेल्स में अभी हम जाएंगे नहीं, because nebulin का role is not much into the contraction part. It is about how the development and कितने length का होता है thin filaments and all those things are the areas where probably nebulin has a role. But otherwise we basically can explain the entire contraction process with the help of this myosin cross bridges, the myosin heads and their interactions with actin, and with the help of troponin and tropomyosin. So that is what essentially we need to know. So you can look into the detailed zoomed in picture कि यही है कि tropomyosin. Tropomyosin is again a filamentous structure. It is also a dimer. It is made up of two polypeptides dimerized, and it will cover, what does it do? Tropomyosin basically cover करते हैं myosin binding sites of actin. तो actin molecules, these individual globes or globular molecules have myosin binding sites. इस myosin binding sites को mask करके रखना tropomyosin का रोल होता है. तो 7 actin molecules के myosin binding sites के उसको mask करके रखते हैं tropomyosin. And tropomyosin interacts with the molecule which is called troponin, and troponin is the molecule which can sense the changes in the calcium levels in the muscle cytoplasm or sarcoplasm. And when the calcium levels are higher, troponin interacts with tropomyosin to expose these sites. ये जो मास्क करके रखा हुआ है उसको expose कर लेंगे जब कैल्शियम बढ़ेंगे, and that is the time when these heads can interact with the actin. Now we will come to this process exactly how it is happening later also. And if you look at the myosin molecule में detail में देखोगे तो myosin is made up of two heavy chains. You can see that alpha helical structures here with emerging out as two heads. So myosin heavy chains and then two pairs of light chains. They are essential light chains and regulatory light chains. So these are two pairs of light chains, essential light chains and regulatory light chains. Now at the head of the myosin, see, you know that contraction of muscle is an energy requiring process. मैकेनिकल वर्क कर रहे हो आप केमिकल एनर्जी को यूज़ करके, तो ऑब्वियसली एनर्जी रिक्वायरिंग प्रोसेस है. तो एनर्जी कहाँ से आएगा? एनर्जी तो एटीपी से आता है. सो विच वन ऑफ दोज मॉलिक्यूल्स विच विल बी इन्वॉल्व्ड इन दिस एंटायर प्रोसेस शुड बी हैविंग एटीपी एज एक्टिविटी, and it is the myosin head which can bind ATP and also hydrolyze it and releasing the energy in the high energy bond. So myosin के head में you have this light chains or ATP binding sites where you have the ATP as activity, and obviously myosin has to interact with actin, so myosin would be having actin binding sites. So light chains में जो essential और regulatory बोलते हैं, essential light chains की presence is important for the ATPase activity. So if essential light chain is not there, then the myosin head's ATPase activity will be lost. So that is why it's called essential. Yeah, meromycin. So enzymatic digestion dekar you can divide the molecule into further. So that I am not going into the details of it. I could see a message on meromycin and other ways of splitting the structure into two fragments and looking at it. But basically for functional understanding, what we need to understand is, okay, we know about myosin and actin cross bridging. Lekin myosin has actin binding site, actin has myosin binding sites which are masked by tropomyosin. Then myosin head has two pairs of light chains, and essential light chains are important for the ATPase activity. तो ATPase activity myosin के head में रहता है और वहाँ पर you also have the ATP binding sites. So that is where you have the ATPase activity, and then you have the tail portion which is in the form of alpha helix, and many such tails interact to form the thick filament. तो एक thick filament अगर लेते हैं तो उसमें many of this thick, the myosin molecules are there. At the towards the M discs you have the tails oriented, and away from the M discs you have the heads oriented, and these are the heads which are going to interact with the actin. And you know, thick filaments are surrounded by thin filaments in a hexagonal manner, if you remember this cross section. So if you look at the A band, each thick filament is surrounded by a thin filament, 6 thin filaments in a hexagonal pattern. So that is how they are going to interact. So these all things we had discussed, and this is the orientation I was talking about. In the center you have tail to tail connection happening, tail to tail, and from here on this side you have head to tail arrangement. मतलब एक हेड के बाद एक टेल आएगा, हेड के बाद एक टेल आएगा. यहाँ पर टेल्स इंटरैक्ट टुगेदर, मतलब दे आर जॉइंड टुगेदर. तो either side you have the bipolar arrangement where the heads are pointing away from the M discs. So that I think would complete the structure of those molecules. Now here you have another term which is called costamers. Now costamers, what do they do? Costamers basically connect the sarcomeres and help in stabilizing them and transmitting the force in a lateral manner. Sorry. Yeah. So see this picture. You have this A band, Z discs, everything. Now this is inside the sarcolemma. Now sarcolemma is a membrane structure. It's a membrane. It's a same membrane which we were talking about in the last classes. So that's made up of lipoproteins, lipids and proteins, and you have you need, see, when the muscles contract, there will be lot of forces. There will be contractile force generation, quite a significant amount of force being generated. The muscle will get deformed. Some amount of force needs to be transmitted laterally also, in the sense कि जब ये सब contraction वगैरह होगा, ये Z डिस्क्स को और M डिस्क्स को स्टेबलाइज करना अल्टीमेटली सेल मेम्ब्रेन से नहीं हो पाएगा. See, these are the zones where the thick and thin filaments are stabilized. Like at the center you have the M discs, here you have the Z discs. Yeah, so I think that's some reply to one of the earlier messages. Yeah, so you have the Z disc and M disc here, and you have the force generated. Now the cell membrane cannot stabilize these zones without the help of extracellular matrix. I hope you understand this, because these are the zones where you have the thick and thin filaments incorporated, and the force they are going to generate when they are doing the crossbred cycling is quite high. So there will be traction, deformation happening in the cell membrane, and cell membrane cannot mechanically stabilize these structures. So what happens is these Z discs as well as the M discs are actually supported through the membrane to the extracellular matrix. Now this is the picture of showing different molecules. Don't get overwhelmed by the names written here. Basically what you need to understand is, this is the sarcolemma. The sarcolemma has integral membrane proteins, okay, these membrane proteins in the form of sarcoglycan, dystroglycan, ये सब नाम है, ये याद रखना ज़रूरी नहीं है. Integral membrane proteins है. Now these are linked to through some adapter proteins to the myofibrils, जैसे यहाँ पर filamentous actin से binding दिखाया. So on one side, inside the cell you have this myofibrils and actin being connected to the integral membrane proteins through some adapter proteins. So that is how it is. Like for example, syntrophin. In this example, syntrophin is an adapter protein which connects filamentous actin to these integral membrane proteins. उसके बाद वहाँ से through some peripheral protein, it is stabilized to the basement lamina proteins and extracellular matrix. मतलब basically ये है कि ये myofibrils, they are not just connected to the cell membrane, because cell membrane cannot stabilize them, because you need a good mechanical support to stabilize Z discs and M discs and all. So these filaments, the myofibrils, are connected through the membrane with the help of adapter proteins and the peripheral proteins outside to the basement lamina protein and extracellular matrix. And extracellular matrix is quite robust and strong, so it can support the muscle extracellular matrix only. It can support these disc proteins and stabilize the myofibrils and help them in proper force generation. So costameres are basically these connections, this entire complex. Costameres are nothing but the protein molecules involved in this entire interaction, mechanical connection between the myofibrils through their individual filaments, through the integral membrane proteins and other proteins to the extracellular matrix. So इसको बोलते हैं costameres. Why we have brought this into picture is, if some of these molecules get dysfunctional because there is a mutation in one of the genes, then you have lot of disorders in the form of genetic disorders which are called muscular dystrophies. This terminology you will be getting more familiar in pathology classes and later in medicine, that what are muscular dystrophies. And muscular dystrophies में dystrophin protein, I don't think dystrophin, you'll have to check. It's somewhere associated in this costamere complex. You can see dystroglycan and sarcoglycan complex here. I haven't labeled all the proteins. This picture doesn't have all the involved proteins. There are many more actually, and probably many of them or few of them are yet to be discovered. But you have many proteins including laminin and many other dystroglycan complex. So we can check dystrophin कहाँ पर है, check करना पड़ेगा. But what is important is, if those proteins in the costamere complex gets mutated, then there could be degeneration of muscle fibers, muscle damage, muscle injury वगैरह होता है, और उसकी वजह से muscle weakness भी होता है. So this is one set of disorders. No, see, fixing doesn't mean that abrupt fixation. It can move. But in the perpendicular direction, they can actually see, when you talk about shortening of sarcomere, there are two types of contractions. One is when the muscle actually shortens. When the muscle shortens, matlab dimensions, say each sarcomere 2.2 micron से say 1.8 micron तक जाता है, तो isn't this move? Those movements are still possible. क्योंकि दिस मॉलिक्यूल्स आर ऑल डिफॉर्मेबल, द डायमेंशंस कैन चेंज, एक्स्ट्रासेलुलर मैट्रिक्स भी यील्ड करते हैं, ऐसे नहीं कि एक रिजिडली फिक्स हुआ है. इट इज पॉसिबल एंड माइक्रोमीटर रेंजेस में मूवमेंट्स आर पॉसिबल, बट they stabilize so that this discs are actually stabilized, otherwise they will just mechanically damage the cell membrane. Cell membrane cannot stabilize them. That doesn't mean that movement is not allowed. Movement is still possible, and this kind of attachment is also responsible for some amount of passive tension in the muscle. So मूवमेंट पॉसिबल है, लेकिन स्टेबिलिटी इंपॉर्टेंट है, वरना एक दो कॉन्ट्रैक्शंस के बाद पूरा टूट जाएगा. ऐसा सिचुएशन नहीं होना चाहिए. एंड सेकेंडली, not all contractions you have the shortening happening. There is a set of type of contraction called isometric contraction, जिसमें the sarcomeres just build up tension, they don't shorten. We will come to that in subsequent classes. So in those contractions actually there is no movement within the sarcomere. It generates tension by some other mechanism. Obviously crossbreed cycling is going on, but sarcomere doesn't shorten. So you have shortening contractions also. So contraction doesn't always indicate there is shortening. Contractions mean there is crossbreed cycling and there is a mechanical interaction, a force generation which is happening. So force generation can happen without shortening, and if shortening is happening, then there will be movement. There will be movement and there will be movement of the joint. So you have the shortening contractions which are called isotonic contractions, and there are contractions in which you don't have shortening or the length of the muscle remains same, which is called isometric contractions. This isotonic and isometric I will be coming tomorrow, tomorrow's class we will be discussing about it. So what I try to convey is that these interactions are for mechanical stability, not that Z discs and M discs are fixed to a particular point. They can move. It's not that it's not possible to move, but they are stabilized with the help of these interactions, so that they can sustain multiple contractions and relaxation cycles. So now when you come to, we had said that these cycles, thank you, these muscle cells are multinucleated. Can anybody tell why are they multinucleated? Because they originate by the fusion of multiple cells. So when the muscles are developing embryonically, so there are cells which are differentiating from the paraxial mesenchyme. Obviously, I think you will learn more on this in embryology classes in anatomy. This is not within the scope of physiology, but what you need to understand is that there are cells called myoblasts, which are the primitive cells, जिससे ये muscle fibers originate करते हैं. So these cells fuse together in a linear fashion. That is why you have the linearly or the long muscle fibers, or some of these blast cells differentiate into satellite cell, which are kind of stem cells. Stem cells मतलब अगर कोई mature muscle fiber damage होता है तो नया muscle fiber can originate from this stem cell. So these satellite cells and the muscle fibers interact closely after differentiation, and since each muscle fiber is formed by the fusion of multiple myoblasts, you have this multinucleated state, and these nuclei are pushed towards the periphery because they are filled with myofibrils. So so the fusion होने के बाद जब muscle fiber बन जाता है, it starts expressing the proteins which are very specific to the muscle, like the thick filament protein, thin filament proteins. ये सब जब express होता है तो mature हो जाता है, and that is a time when the nucleus will be pushed to the sides. So with this we come to the basic structure. Now I will also give you the introduction to the next step of contraction. So we just introduced you, I just introduced you the structure of the muscle, कि क्या क्या होता है muscle fiber को. If you cut open and see, satellite cells, as I said, they have many roles. Actually they are the stem cells. Stem cells मतलब most of the tissues have some kind of stem cells. Stem cells are the cells which can actually differentiate and become the mature cell when there is a requirement. That's what I was saying. When a muscle fiber is injured, then the satellite cells can actually differentiate and become the it can stimulate the formation of, yeah, they are also derived from myoblasts. Yeah, I'll just check into this once again. I just see we don't go into the details of, there are many things which satellite cells actually do. Earlier we had conducted a seminar also for undergraduates on what are the roles of satellite cells and all. So you can look into this. but as I said, they are the resident stem cells. So every tissue, even in your skin, you would have some of the cells which are there for renewal function. Renewal function का मतलब होता है कि अगर कोई mature cell डैमेज हो जाता है, you need to replenish them. For example, intestinal mucosa. Intestinal cells are the cells where you have rapid turnover of cell. Turnover क्योंकि mucosal cells damage हो जाता है in the process and you need to replace them continuously. Muscle cells don't have that much high turnover because muscle cells don't get damaged that way. But when you are doing, so for somebody who's going for strength training exercises, in physical conditioning के लिए, so when you do exercise to that level, some of these muscle fibers actually get injured, but they get replaced by new cells, and that regeneration is possible, especially in the muscle cells. So that is with the help of the satellite cells. We can look into a little more details. I can come up with more details maybe in the next class. So, but that is not something which is very relevant to you at this moment. We just need to focus on how muscle contracts in response to the stimuli. But you are free to read about satellite cells and their role for your own understanding. Okay. So coming to excitation contraction coupling. Now, as the terminology itself says, कि muscle की तरफ excitation आ रहा है. Excitation मतलब it's coming from the nerve and neuromuscular transmission की वजह से the signal has crossed from the nerve to the muscle, and in the muscle you have an action potential being generated the same way an action potential is generated in the nerve. You have voltage gated sodium channels and potassium channels playing the role. Now muscle membrane में action potential आ गया है, लेकिन ये action potential is an electrical signal. Now you have to make that electrical signal trigger the mechanical work, or it has to stimulate the muscle to contract. Now this excitation, how is it coupled to contraction? What are the mechanisms involved in this coupling of excitation in the form of an action potential on the muscle fiber or muscle membrane, sarcolemma? उसको आप लोग उसको कैसे couple करेंगे mechanical contraction के साथ? ये this is a question and this is an important mechanism which we need to understand. ये कैसे होता है? And that is what is excitation contraction coupling. So neuromuscular transmission and genesis of muscle action potential that spreads along the sarcolemma, उसको बोलते हैं excitation, and contraction is the shortening or force generation by muscle, and you need to have a coupling between these two. So this is a graphical representation of the muscle fiber की action potential. So muscle fiber muscle excite हो गया, neuromuscular transmission के बाद muscle के ऊपर एक action potential generate हो गया. So वो action potential यहाँ पर है, and this will be propagated along the sarcolemma. उसके बाद you have, after a time delay which is called the latent period, a latent period is there, a latency is there. उसके बाद muscle जो force generate करते हैं, this y axis is muscle fiber tension, it increases, reaches a peak which is the peak of the contraction. उसके बाद muscle relax करते हैं जब tension घट जाता है or comes back to the original state. So this is excitation, this is contraction, there is a latent period, a latency, which means something is happening here, some chemical reactions are happening, some changes are happening, which takes some time. After the excitation is done, there is some time required before the muscle can start generating the force. So whatever you are going to look at is happening in this time window, which is the latent period. So ये coupling कैसे होता है, and who is responsible for it, which is the ion, or which is the chemical agent, or what is the mechanism, how is it happening? This is something which we need to do, which need to understand. Now scientists were looking what could be the mechanism, why is the muscle contracting? Obviously there is some thick and thin filament interaction, और ये कैसा होता है, what is triggering it? These are all were the questions in the minds of the people who were investigating it. What we came up was, before the muscle contracts and after the excitation is done in the form of action potential in the muscle fiber, what people could characteristically see was the rise in the level of calcium in the myoplasm or the sarcoplasm. So जब muscle fiber में action potential हो जाता है sarcolemma में, उसके बाद calcium की level बढ़ जाते हैं myoplasm में, and the force generation follows the rise in the calcium level. So after the calcium starts rising, you have the force generation also happening. The peak of the calcium comes much earlier than the peak of the contraction. There is some delay here. and then the peak of the calcium drops, it comes back to the original level, the tension also comes back to the original level, but there is a delay. So it seems like muscle action potential triggers the rise in calcium, and the calcium is probably responsible for the contraction. So this is based on the temporal relationship. When you have a temporal sequence like this, we tend to believe that okay, then it seems like muscle fiber action potential might be triggering calcium release, and calcium release might be relating to the contraction. Now people did lot of experiments to prove that the rise in calcium is required for the contractions to happen. If you don't have calcium rise in the sarcoplasm, even if you fire an action potential in the muscle membrane, you won't have a rise in force, you don't have a contraction if you don't allow calcium to rise in the sarcoplasm. So that cause effect relationship has been established by different experiments, and now we need to know कि जब muscle fiber में action potential होता है, so latent period is after action potential or during it, and latent period you count from the point of stimulus, so action potential is also counted within that. So action potential is also included, muscle action potential is also included inside the latent period. Yeah, start of the action potential से है, तो action potential की duration भी इसके अंदर count होगा. So now we need to know how action potential will lead to rise in myoplasmic calcium. Then we will look at how rise in calcium is responsible for the muscle contraction. So with this, I will wind up for today. Tomorrow we have 2 classes. So I will be covering excitation contraction coupling, and then we will move on to the mechanical aspects of contraction and some other aspects as well. So, and then Saturday afternoon, please be aware that we have the case based learning, and be prepared with the content which I have already shared with you. That is very important, and it will be an interactive and interesting session because you will be exposed to a clinical case, and I am sure that that's something which you would be looking forward to. That is what ultimately you have to do. So you are going to be all, most of you will be actually becoming physicians in future. So that is what you are supposed to do. Look at clinical cases, clinical pictures, try to analyze it and make some meaningful interpretations. So we'll be doing that on Saturday afternoon. So with this, I wind up today's class, and I will send you the slides. I have not right now. I have not sent the slides. I will send you after tomorrow's class also get over. I will send you the entire block of slides till the tomorrow's class. I will send you. But right now, I have not sent it primarily for one reason. I would want you to go through primarily through one of the textbooks which you would like to read. तो अभी slides में ज़्यादा concentrate मत करो. Even for the exams, I am not particular about what you write, whether it is given in my slides or whether it is my slide content or anything. Look, that's not the point. You need to understand the concepts, and reading a textbook perhaps will do better at this moment. So go through any textbook, take one textbook of your choice which you would find easy to read and understand. Discuss with your friends. If you don't understand, clarify some of the queries at your end itself, and read the chapters, read page to page, and that is what you have to build up the understanding. Exam ki time mein maybe going through the slides and looking at the pictures and all might help, but most of these important pictures jitne bhi maine dikhaya hai, most of these are available on the textbooks also, and if it is not available in the textbook, it's not very relevant as of now. I am showing some of the pictures only for your understanding, which is desirable but not essential. So right now focus on the essential things. You can, once your essential understanding, basic understanding is clear, then you can build upon that. अभी क्या है ना, ज़्यादा इन्फॉर्मेशन से ओवरलोड हो जाएंगे तो मुश्किल हो जाता है इनिशियली. So stick to the basics, whatever is given in textbooks, focus on that right now. We can further expand on this once you have your basic understanding clear. I hope that is fine, and ठीक है. So anything more?
How we have to read the approach to motor disorder? There are 96 pages in the PDF. We have to read all of them.
No, the approach to neuromuscular disorders, I have given you some keywords in that preparatory material, like differentiating between lower motor neuron and upper motor neuron disorders. Okay, and I think I think there are a few more keywords. So what you do is that, and that 96-page document, within the PDF file, you search with these keywords, okay, and where you find those keywords, just read those portions only. That's one way of that is what, because see, if I could, though I could have extracted the essential part only, but then that will lose the continuity. So you focus on the keywords which I have mentioned in the email which I had sent you. उस keywords के around पढ़ना होगा. And even if you don't understand, even if you don't complete also, no problem. On the day of the session also, you will be having access to this material. You can search that time also, pointed queries के लिए. So, but this is one of the sources which we found very useful, so that you can read it and understand. ठीक है, तो keywords के around पढ़ लेना. I hope that will be okay, and thank you for your patient listening. Thank you. डेस्कटॉप पे है। अगर जैसे बुलाना चाहें तो कहाँ बैठते हैं, कहाँ बैठते हैं अगर बाई चांस बुलाना है किसी वजह से नीचे? हाँ आपका कॉन्टैक्ट मन जी। अपने आप शुरू कर दूँ 2 बजे?
हाँ चलो
सब रेडी है। You are more interested in surgery now. So then you took me against signal again. So how much did you get that time?
I got a rank of 150 there.
For the backlog?
150. But I was always interested in research, so I passed. And then I realized that's not in your competition good, so I gave the exam again. This time I got 1000 and I took the first one. Even the first time I was confused between biochem and parcel.
When was the first 1.5 to 2018?
2019 January I joined there, and in 2020 June I resigned.
2019, yeah, 2018. You were there for June. Where are you from?
I'm from Visakhapatnam, Andhra Pradesh.
Oh, you. I stayed in Vizag for long.
Oh,
I spent half my life.
okay. I was from Andhra Medical College. Yes.
So then you wouldn't have come for that physician scientist thing which is continuously held.
I don't know.
That time. In fact, one of the years Asta, yeah, she's there. She was there when I spoke. Okay, so you were part of that discussion. See, I spoke towards the end.
Last,
You were there.
that was Sunday, no sir. Last में Sunday
Yeah.
की थी.
No, because I remember maybe because it was my meeting was towards the end.
I remember sir. Yours was the last one.
It was supposed to be at the beginning, 4:30, but then somehow it got rescheduled and it ended up being at 6 o'clock. There were really few people there. We were around five.
Actually, I myself am very much interested in this sector because if I get into wet lab work, I'll be restricted to the institute. I should stay in the institute where the setup is. But if I can get into the work that you are, then I
Yeah,
can.
then it is independent.
Sir. So I have a little more freedom.
Lot of freedom, not very Most of my work is on the paper and on the computer. That doesn't matter where I am.
Sir.
Independent of that. That has its stuff, but it has its own ifs and buts and happens. Yeah, sure.
Good afternoon, everyone. Yeah, okay. So I think we'll start. Yeah, good afternoon, everyone. I think we'll start, right? Today is a longish kind of lecture. How, like I described, okay, so we'll take it from Monday. So today I'll talk about a few specific things. I'll try and balance what is important for you insofar as the exams go, and I'll also try and balance something that involves a little more than that, something that's also a little interesting for you all to. But there will be a lot of gaps that you'll have to plug in by yourself. What I would suggest, because it's a longish kind of lecture, take notes on what is it that you don't understand right now. Okay, and either we get a little time after the lecture or maybe at a different point in time, but we'll have enough time at some time where, but at least if you had your questions noted down, it'll be easier to discuss that. Okay, and anyway, you're free to interact with the faculty whenever you all want. Okay, so try, and unless you're really confused about something, just make a note of whatever you don't understand, and we'll try and talk about it. Okay, but like I said, this is a very important topic, and from your understanding as a medical student, as well as research, and as well as your exams. All right, so this is enzymology. A part of enzymes has already been covered. Okay, so I won't kind of skip that. I'll just go through these PPTs. Many of them probably you've already seen, but I'll come to kinetics straight away, but I won't ignore it. Okay, so I'll give you all about a couple of seconds on each slide so that you all can make a note and figure out as to if you need to discuss this further. All right. Okay, so first we'll talk about, as we discussed that time, what are really enzymes. Okay, so enzymes are nothing but something that increases the rate of a biochemical reaction, and as we talked about last time, they're not just proteins. Okay, so you have RNA like we described, there are DNA enzymes as well, and there are also what are called abzymes or catalytic antibodies. All right. Okay, so for this particular lecture, we'll focus on proteins as enzymes rather than RNA or DNA. All right. So most enzymes typically are what are called proteins or globular proteins, and they have a wide range. Okay, they're made up of amino acids. So you can have an enzyme that is as short as 64 amino acids, and you have something that's absolutely large, like what is called a fatty acid synthase. Okay, so these are some of the important properties of an enzyme. Okay, they're not consumed by the reaction, they really do not affect the equilibrium. Okay, and their action is very specific, and what defines an enzyme is something called its active site. Okay, so what is an active site for an enzyme? An active site is nothing but a 3D representation of something, a part of the enzyme that participates directly in catalysis. Okay, again, what is an active site? You'll have done, I think, proteins, various orders, right? You've done primary structure, secondary structure, tertiary and quaternary structure. From that understanding, since enzymes are proteins, the ones that we're talking about here, it's easy to understand that when proteins kind of fold into their they bring large parts of polypeptides together. Now what happens when they do that is they create kind of pockets, clefts, pockets, grooves. Okay, now these are not present as such in the primary structure, but they'll start becoming visible as these proteins fold. It's in these sites that catalysis usually takes place. So this is what is the active site of a particular enzyme. Okay, so I'll discuss that a little more in detail. what you'll need to know is one of the important things that you will come across in enzyme biology is something called this number. It's the enzyme commission number, okay, and it's there in every book. Wherever you see an enzyme, invariably you're going to see this in brackets, okay, this set of numbers. So what are these numbers? This is just again for your understanding of enzyme biochemistry, all right. So A, B, C, D, you have class, subclass, which is represented by the donor molecule, and something called a subclass, which is the acceptor, and an ordered list. In other words, in order of discovery. Okay, so you can have something, okay, we'll come to that. Okay, so this is what is called the EC number of an enzyme. Okay, so and if this is something that you absolutely need to know. Okay, so enzymes which you'll have done before are categorized into these 6 reactions. This is an important part. You need to understand this, you need to have examples, and you need to understand what is the difference between some of them. Okay, so the standard classes are oxidoreductases, then you have the transferases, then you have a difference, you have hydrolases and lyases. So you frequently ask the difference between these two. Basically it involves water, and in lyases you basically there's no water involved. Okay, then of course you have isomerases and you have what are called ligases. So this is the standard classification of enzymes, which with examples you'll absolutely need to know this. Okay, all right, so this is one of the topics that we fall back upon. Okay, so it's as important as that. So let me give this to you as an example. Okay, so let's go back over here. As you can see, isomerases are number 5 over here. Okay, so I've basically shown this to you, and everything that I'm going to show you is color coded, so it's easy to understand. All right, so isomerases class 5. All right, in this you can see that there are many subclasses. Okay, so you have 1, 2, 3, 4, 5, and it actually goes all the way up to 99 sometimes. All right, so let's go step by step. So the red one is either a racemase or an epimerase. Leave the terminology aside. The important thing right now is to understand it. All right, so class 1, that can be subdivided into whether there is a donor or an acceptor. So if it involves amino acids and its derivatives, it's number 1, which is coded in green. If it involves carbohydrate and its derivatives, it's in blue, which is coded in blue. Okay, I think this is changed color, I'm sorry. Okay, I don't know why this is changed color, I'm sorry. Okay, this number 3 should be blue. Okay, I don't know why it's changed color, but anyway. Okay, so amino acids and its derivatives, so you can see racemase. Okay, so isomerase, racemase, amino acid, and the order of discovery. On the other hand, take a look at the other ones, the ones that involve carbohydrates. Again, isomerase, racemase, but it involves carbohydrates and its derivatives, which are number 3. Okay, so this is for you all to understand how it is. So then there are some very specific examples. This is an important enzyme that you all will come across. As you can see, this is a full reaction. Okay, this is a reaction that's catalyzed by this enzyme. Okay, so this is its oxidoreductase, okay, which is class 1. It has a donor, so this subclass and subclass are based on donor and acceptor, and it's 49th in order of discovery or characterization. Okay, so this is how it's broken up. Okay, these are some more examples. I think we can skip this part, but pretty much along the same lines. Okay, all right. Okay, so now let's come back to what we described is the active site geometry of an enzyme. Okay, so like I said, this is that part of an enzyme which actually does not exist technically. It only comes together when an enzyme gets folded. Okay, so this is what is responsible for the particular reaction, its specificity and its efficiency. Okay, and as we talked about, it creates certain things on the enzyme, a pocket, a cleft, a groove, and so on and so forth. Now what does this do insofar as when you're talking of it as an enzyme? The first of all, it shields the substrate from solvent. Okay, in other words, it allows, as you can see, the natural consequence of this, of excluding the solvent, is that the residues or the amino acids of that particular enzyme are free to interact with that particular substrate. You get my point? It's an enzyme, right? It's a protein, it's folded. When it comes across a substrate, okay, because of the active site geometry, you're actually excluding the solvent away from it. As a result, amino acids which constitute the enzyme are free to interact with the substrate. And as you all have already understood, amino acids are not very passive molecules. Many of them have very active groups. Okay, for instance, the hydroxyl groups, the thiol groups, okay, these are all very much present in amino acids. So many of them are electrophilic, nucleophilic, and when they interact with substrates, it creates a reaction. All right, so that's what it means. That's why we are talking of active site over here. There are some other very important properties of active sites, okay, and that is, one is that out of all the enzymes that are over there, all the amino acids, you will probably find that the active site geometry is restricted to very few amino acids. Okay, so the rest of it are more called what are called structural amino acids, which are important for folding and maintaining the integrity of the enzyme per se. But as such, the catalytically competent amino acids are very few in number. Okay, and you can actually count them on your hand. Okay, they'll be members of the acid group, they'll be members of the basic group, they'll be hydroxyl groups, thiol groups, and so on and so forth. Okay, what is interesting to understand is that many of these active sites are conserved. Can anyone tell me, what do you understand by conservation? You all have heard of conservation in so far, as the environment should be conserved and so on and so forth. But what are we talking about over here?
Through generations.
Through generations. You'll have to speak up. I'm hearing 2-3 voices at the same time. Yeah, go ahead.
Keep same across time.
It's the same.
It's the same, but it means what? Same where?
Same in microbes and human beings.
Okay, fair enough. I think yes. Okay, so that is actually what it sums up. Because now see, when you're talking of catalytically competent enzymes, when you're talking of conservation, you're talking conservation in terms of evolution. All right, which basically means the same enzyme. Okay, now listen to this very carefully. The same enzyme that is present in human beings will be present in different organisms. The proteins will differ, no doubt, but what is very nice to understand is that many of these amino acids, which are actually catalytically competent, are conserved across species and organisms. Which means if you look at the proteins together, you'll be surprised that many enzymes, okay, especially at their active sites, have exactly the same amino acid or something which is close to it. That is what is very important insofar as enzyme biochemistry goes, because it can give you a way. What is the use of it? I was going to give you the answer, then I thought I'll ask you only. What is the use of it?
Recombinant protein formation.
Recombinant, fair enough. Anything else? What's the use of it?
Tracing out the evolutionary patterns.
Fair enough, but use. Let us say it's conserved. What does this mean? We can make.
If the active site is conserved, then if introduced into the body of another species, it will not, manifest any immunological effects. So that can be one application of this.
Okay, but I see we are talking of the ones which are conserved, not the ones which are different. And these are very few amino acids.
Use in the sense human advantage or by nature has kept this trend.
Yes, by nature it has kept it, and therefore it also helps us do something very nice. It helps us identify possible enzymes or enzyme analogues in different organisms. If you know the activity of one particular enzyme and can characterize it to a few amino acids, you can by a series of computational tools pull out similar amino acids in various other organisms and actually pull them out in the laboratory to study them. In other words, these give you a way to understand other organisms and their enzyme biochemistry as well. Alright, I'll give you an example. Okay, so this we can leave. Yeah. Okay, so just very quickly, I'm going to show you a few pictures in terms of what an active site is. Alright. This is the active site, or this is what is called the ball and stick model of lysozyme. Okay, where is lysozyme present? Yes, absolutely. It's a very common enzyme. Okay, it's a very small enzyme which is about 129 amino acids, and the beauty of this enzyme is it's very well conserved across species and organisms. So as I can show you over here, yeah, so as you can see, this is the three-dimensional structure of lysozyme, and as you can see very nicely, you see this part over here, you see this? This is what is called the substrate cleft or the pocket that this three-dimensional structure forms. So let me take you again, okay, here. So if I were to have the same enzyme, but you see this? This is the cleft or the substrate pocket. What it will do? There you are. You see this? This is how enzymes work. So you see, we started off with this one. This is what it looks like, and this is its substrate. Now, because it's folded in this kind of way, what you'll see is that this is formed, and this is where the substrate fits. Alright. So this is the example of an active site. Okay. So what is it really made up of? So you'll have done the structure of proteins, alpha helices, beta strands, etc. Yeah?
Yes, sir.
Right. Now I'm going to show you in this diagram how you can all put that secondary structure together in the form of the active site. Remember, this is the same image. This is the same image with different things which I am highlighting. So if I take a picture of this, which you're seeing over here, with the substrate over here, I can change a few buttons and convert this amino acid structure into something that is a mixture of alpha helices and beta strands. Alright. Let me show this to you. So I convert this image into this. It's exactly the same image. No difference at all. But what you're seeing is, you see the active site geometry over here. It's made up of these 4 residues. Just 4 amino acids. Glutamic acid, okay, at 2 places, aspartic acid and an asparagine. And as you can see, this is an alpha helix, this is a beta strand, and this is a coil. So exactly the same thing. So it is this set of amino acids, 35, 57, 52 and asparagine at 46, it is what is conserved across organisms. Okay, maybe the protein will differ, don't get me wrong, but it is this which is conserved. Alright. So like this one, and this is nothing but a blow up of the same thing. So it's the same image again, 35, 57, 52, 46. So this is an even close up version of the same diagram. Alright, showing you this is the substrate of lysozyme and this is the amino acids which are actually interacting with this. So you see this? This is what an active site is, and this is how catalysis takes place. Because there is no water around, the amino acids of the protein are free to interact. So in fact, this red atom is which? Any guesses? Anyone? Yeah, good. It's a group, yeah, but it's actually the oxygen one. Usually in these diagrams they don't show hydrogen. All right, so the red one is actually oxygen. Okay, and this one is usually sometimes they show it as nitrogen. All right, good. Okay, so that leads us to this part. We were talking about conservation. These few residues, now this is lysozyme from different organisms. This is lysozyme, the same protein from different organisms. Okay, and you represent them in either a phylogram or what is called a cladogram. But this is what is interesting, because if you'll see, essentially the same amino acids are present everywhere. Okay, so with very, very subtle differences, you'll find this 49, 42 and all that. You see this, these conservation. So different organisms, but similar amino acids. Okay, so that's the importance of conservation, where you can actually find amino acids. Okay, well, I'll have to jot this down. Okay, wait, how does an active site shield it? Okay, because in many ways the active site is folding. Remember, when the protein is in solution, the protein is folding. Okay, so once the protein starts folding, there are certain regions which are excluded from solvent. Okay, see, it's not, you've done that, right? As long as it's a primary structure, that's not a problem. But the moment it starts folding, you're going to have areas in between those folds which are completely excluded from the surrounding solvent. So those areas have very, very specific terms in enzymology, and like I said, they are either pockets, they are cavities, they are grooves, they are subgrooves, and so on and so forth. So these form when a particular protein is folding. It will always form. Okay, as there are several software that are able to actually predict which and how many regions are actually excluded from the solvent. As a result, like you see, like in this example, when such an, for instance, this particular one, okay, here what will happen is, so this is a kind of a surface groove, but I haven't shown you over here. You'll find many small pockets over here which are excluded. I haven't got that diagram with me, maybe I'll show it to you some other time. But this particular enzyme, this will probably be the biggest cleft, but there'll be multiple smaller ones over here also, which obviously will be unsuitable for a substrate. Okay, because this is a big one, therefore it has to fit here. But you might have possibly smaller substrates that also fit in this. So in many ways, protein folding excludes the solvent and gives an environment where the amino acids can actually interact with that particular substrate. Okay, you'll have to do a little visualization in all this. Okay, all right, so let's move on. So this in so far as all this we've described as conservation, and basically this is phylogenetic analysis of lysozyme across different organisms, okay, which shows the conservation in amino acids. All right, then very quickly we'll talk about catalytic mechanisms. I won't go into this because I don't think this is part of your topic today, but basically enzymes work by proximity, induced strain, something called acid-base catalysis, and covalent catalysis. Okay, so these are the four very specialized ways. Okay, more for a teaching purpose than anything else. There are many overlapping ones also, but this is just for this. So you should know this because many a times we ask you this question. Okay, and this example is a very important example. Okay, proton shuttle and the charge relay network. All right. Okay, so again I'm going to skip through this. This is just to show things to you. In case you want to know a little more, you can always ask me later on. Okay, so these are some of the mechanisms. Okay, so by induced strain, wait, we'll have to come to that, just a second. So one of the questions I shall answer is, if they are the same sequence, why don't we have separate names? No, separate names is a different aspect altogether. The enzyme has a certain set of characteristics. We can say it's an enzyme, it's a lysozyme from mouse, it's a lysozyme from humans, but effectively it has enzymatic activity to the same set of substrates. So it doesn't matter, the name doesn't matter. We just say that the organism differs, no? So if it is conserved and has, yes, it's a lysozyme, but from a different organism, from 20 different organisms. Okay, so you'll have to say, in fact, when you go to databases, you'll check human lysozyme, mouse lysozyme, rat lysozyme, that's how it is. Okay, so that's why it is there. So the lysozyme part will remain the same, but obviously the organism will differ. Okay. All right, this is just again more of the mechanism. We'll skip this. All right. So then we come to a very, very important concept, the concept of allostery. Okay. So this, of course, was coined by Jacques Monod. Okay, and you also know him for what? Anything else? Yep, that's absolutely correct. The same. Yeah, so it's the same gentleman who did a lot of work on allosteric mechanisms and regulation of enzymes, and therefore he's known for this. Okay. Okay, so what we're going to talk about, what does allostery mean? Okay, so allostery means other. Okay, so anything that involves other than is allo. Okay, so when you have allosteric enzymes, so this is a very, very specific terminology which you must understand. So there's allosteric enzymes, there is allosteric kinetics. Okay, this is something that you need to understand. Okay, so these are those whose activity may be modified by the presence of effectors at a site other than the active site. The key word is at a site other than the active site. So, like I said, an enzyme has, and one more thing you must understand, an enzyme may not have only one active site. It is possible that an enzyme might have 2 or more active sites. Okay, so allostery means anything that modifies the enzymatic activity which is other than the active site or the bona fide active site is called as an allosteric enzyme. All right, so then these are very specific terminologies. So as you can see, there are some terms over here. Some things, it may be the substrate itself or it may be heterotropic. Okay. Okay, so we'll discuss allosteric kinetics a little later. Okay, so this is more of allostery as an example. Cofactors, I think you'll have covered. Okay. So now with this kind of background, we'll start kinetics. Okay. So what is enzyme kinetics? Why is it important and why do you need to understand it? Okay. So like I said, it's the study of chemical reactions essentially catalyzed by enzymes. Okay. When we talk of kinetics, we are usually referring to 2 types of kinetics. Okay. One is what is called the standard Michaelis-Menten kinetics, and the other is non-Michaelis-Menten or sometimes allosteric kinetics. Okay, Michaelis-Menten and non-Michaelis-Menten or allosteric kinetics. Okay. Okay, so what is very important to understand kinetics is there is a lot of terminology involved over here. Lots and lots of stuff that you need to know, because that's how you differentiate between the 2 types of kinetics. All right. So typically, probably I might show a graph to you that's probably better. But anyway, okay, fine. I'll get back to it. So like I said, a typical graph is between the substrate and the velocity of the reaction. Okay. So usually what you plot is the burst phase and what is called the progressive stage. Okay. Most important assumption in enzyme kinetics is something that forms the ES, enzyme substrate complex. Okay, so whenever you're talking about enzyme kinetics, the enzyme substrate complex is fundamental to understanding this. Okay, it is an assumption. It is an assumption which is based on a lot of other groundwork, but a lot of people use that. Okay, so this is that state. So what it says, which we'll study. Okay, then you have what is called steady state kinetics, which means functionals throughout the enzyme. Okay. All right. So let's see what some of this is all about. Okay. Again, this is color coded, so it's easy to understand. So R, which is essentially the rate of the reaction. A, which is obviously your substrate. Okay. Or in many ways your reactant. Okay, classic enzyme kinetics is based on evaluating the concentration of the reactants. Okay, so in this case, this is the reactant. Can anyone tell me why have I used these square brackets over here?
Indicate that it is the concentration of that particular substrate.
Not just concentration, but it is the molar concentration. Okay, which basically means you have to know your Avogadro's number and all those things in your final calculations. Okay, but yes, it involves the molar concentration of these particular substrates. All right, then this one over here is what is called the order of the reaction. In this case is with respect to A. Okay, you put it all together, the green one or A is your rate constant. Okay, simple enough. Rate of the reaction, molar concentration of the reactant, the order, and the rate constant of a reaction. Okay, let's see how we can use this further. Okay, so now let's go through, like I told you, you may not do it all right now, but at least make a note of it that these are some of the definitions which are important. Okay, what is the rate of a reaction? Rate of the reaction is nothing but the amount of reactant consumed or product formed per unit time. Okay, velocity of the reaction, rate at a particular instant of time. Turnover number, a very important part of these definitions. So it is the maximum number of molecules that a substrate can be converted into the product if everything, all the catalytic sites were saturated with the substrate. In other words, it's the absolute maximum that a possible enzyme can do. Okay, so this is the definition of what is called the turnover number or the Kcat number. So as you can see, Vmax, which is the maximal velocity over the total amount of enzyme, especially their active sites are all filled to capacity or saturated with the substrate. Okay, you put this all together, then you have what is basically called the rate equation. Okay, we've talked about this. The order of the reaction is with reference to a particular reactant. This is its formal definition. The order of a reaction is the sum of all the orders of the molecules undergoing a chemical change. To highlight this, I'll show you a small graph which should make it clear. Okay, so this is typically what you normally see. Okay, for instance, if A is a reactant and there are some molecules of it, okay, which is converted into a product, this is the rate, this is its order. If there are two or more reactants with their own stoichiometries, as it's called, this is the order. Okay, and if, as you can see, there are many reactants, then this is the order of the reaction. Is this clear? So you see this order of the reaction is with respect to the reactants. Okay, the other important thing, wait, is here, is understanding the order in terms of functionality. So if I say there's an order of 0, what it means is that the rate of the reaction does not depend on the substrate concentration. Okay, it does not depend on the substrate concentration. On the other hand, as you can see, you have rate 1, there's a single reactant, then order can be 2, there are two variants of this. Either it can be one reactant of stoichiometry 2 or two reactants of stoichiometry 1, right? If I go back to this, like this one, for instance. Okay, so either this can be A plus B involving two reactants, or it can be A raised to the power 2. Okay, two molecules of A. Okay, so that's what basically this means. And of course, if it goes a little more complex, this is what it is. Okay, all right. So when we are talking about models of substrate binding, two things come to our mind. One is what is called a lock and key, which is the Fisher model that you'll have done, which anyway I think is now, it's pretty well established that that's not really going to happen. The key is Daniel Koshland's induced fit model, which gives the protein structure a lot more plasticity when it deals with substrates. Okay, so this is something that is probably the more than rule rather than the exception anymore. Okay, all right, then we come down to, now this is a very very important question for you. You will have to distinguish between Michaelis-Menten kinetics and allosteric kinetics. Okay, and how are you going to distinguish between them is when you're going to describe each of them and their assumptions. So therefore you need to understand what are the points that you are going to use to compare kinetics and allosteric kinetics. Okay, so this, let me explain this reaction to you. This is the basis for everything. Enzyme substrate is converted into what is called the enzyme substrate complex, which eventually gets converted into an enzyme and product. Okay, typically, and this is a reversible reaction, so you can see these two arrows. This is how you typically start represent. This is not kinetics. This is the general scheme of how we are going to work forward. Okay, so as you can see, this is the rate constant for this reaction. This is the rate constant for the backward reaction. That means if ES decomposes into E plus S, this is the rate constant. All right. Similarly, for the other way around. Okay, if ES is breaking up, K2, if E plus T, then E minus 2. Okay, K E minus 2. All right. So when we are talking of Michaelis-Menten kinetics, what are the assumptions? The first assumption is this part. In that, as you see the difference between the two, do you see the difference? So the first and most important difference is that in Michaelis-Menten kinetics, the assumption is that at the beginning of the reaction, little or no product is formed. Little or no product is formed. If no product is formed, okay, very little, obviously K minus 2 is not going to happen, correct? K minus 2, which involves this backward reaction, is not going to happen. So that is a fundamental assumption of kinetics, that at that point of time, very little to no product is formed. Okay, these are some of the other important understandings, the assumptions of kinetics. Okay, so you need to know this. Okay, so we'll talk about that in detail. One is, of course, product formation is then obviously linear with time. As you increase the amount of time, more and more product is going to form. At a steady state, your forward and backward reactions are equal. There is no cooperativity. There is no cooperativity in these assumptions. All right, and this is important because it involves two very important definitions: the concept of Km and the concept of Kd, which you are asked. Okay, usually a single enzyme is important, as we have already talked about. There's very little product formation, and these are some of the other enzymes. Okay, let's see. Okay, now don't panic. Okay, there's nothing and no reason to panic. It's just for your understanding, and sometimes you are asked very very short questions on definitions. Okay, so this essentially is to help you try and understand how you get those definitions and what is the meaning of those definitions. Okay, so let's go with kinetics. Key assumption: enzyme substrate complex is formed. Another key assumption: no K minus 2. So when we are talking of steady state, the rate of forward and backward reactions are the same or equal. That basically defines some of these. That is, RF is the rate of the forward reaction. So as you see, K1 ES. How did we get K1 ES? How did we get K1 ES for this reaction? Remember, the rate is dependent on the what? Reactants. So K1 ES. Similarly, RB which is the backward reaction for this one, is split into two. You can see this, it's either split here and it is split here. So this will be, so this is the reactant in this case, okay. So it's either K minus 1 ES and K2 E minus. Is this clear?
Yes, sir.
Okay. So we are talking about the breakup and formation of the enzyme substrate complex, assumption. What is most important from this is this particular Km. This Km is called the Michaelis-Menten or the Michaelis constant. Okay. You are asked this question, define and describe the Michaelis constant. So in your definition, you will have to show some of this and also say, as you can see, it is nothing but a ratio of different rate constants. So Km is a mixture of K2, K minus 1 and K. Remember that. It has many other definitions. Okay. Km we are going to come across, and you are going to deal with Km very, very frequently. So you must understand what Km is. Okay. It's nothing but a mix and match, an expression, a mathematical expression of various rate constants. Okay. How do these two differ? Let me ask you this. You can see how I've got Km and how I've got Kd. But can anyone tell me what is the meaning of this? I have eliminated K2, so on and so forth. So therefore I have converted this expression into K minus 1 by K1, which is Kd. That's pretty simple and straightforward to understand, right? Can you think of a reason why we are talking about this difference? What is the meaning of this?
Enzyme substrate complex is very stable.
Enzyme substrate.
Equilibrium.
Now see, enzyme substrate complex being stable or not stable, ultimately, okay, fair enough. But okay, I'll take you on that. Wait, let's get some more answers. Okay, what else? One by I didn't get anything.
Converting the enzyme substrate complex into product is way less than its breaking ability to go back into separate enzyme and substrate.
Okay, fair enough. That's what it also means, that ES is really stable. What else? That I can understand from this.
Inhibited from activity. Its activity is not there, so ES won't be converted into ENP. So K2 can be taken as.
Oh, that is also a good answer. Now, where are we driving to all this? Can anyone think of the inference? Let me tell you that all of you all are correct. It is very stable. It probably involves very little product formation. But where is this all leading to? If it does not involve product formation, just a second. If it does not involve product formation, then by definition it's not an enzyme. Yes or no?
Yes.
You get my point. If it is so stable so as to give K2 is equal to 0, then is it an enzyme? Did you understand my question?
Yes.
Let's go to infinity. Let's forget about that. ES is really stable, so stable that it doesn't decompose into E plus P. Is E an enzyme then?
No, sir.
Exactly.
No, sir, it would be acting more like a chelating agent.
Absolutely.
Rather than an enzyme.
Right. And that is the key difference to understand between Km and Kd. That if you are removing K2 from this equation, you are not dealing with an enzyme anymore. You are simply dealing with a complex that comes together and dissociates at some point of time. There is no product formation. It is no longer an enzyme, which means Kd is used to describe reactions when there are two molecules that come together but not necessarily involves an enzymatic conversion. Is this clear?
Can we say KM is real? It is just
Sorry?
assumption.
Go ahead.
KM is realistic one and KD is just assumption. To understand this.
No. They are two different things. They are two different things but along the same spectrum. You are absolutely correct. What if an enzyme simply stopped having K2? By definition, it is no longer an enzyme. So, in principle, you describe KD for two molecules which don't have the ability to convert one to the other. It's an association and dissociation. Clear? There is no product formation. Product formation, you understand that a substrate is taken and changed into something else altogether. That is what catalysis is. Product means what? If the product was exactly the same as a substrate, correct? That would not really work, no? We want a product which is distinct from the substrate. That's what catalysis is all about. So if you're not going to have catalysis, you're not really going to have an enzyme. So you must understand this very, very clearly. Km involves an enzyme, but you're right, Kd involves also a complex of two reactants, but they are not enzymes. This is not an enzymatic reaction. Okay, this is very, very important to understand. So Kd describes what is called association or dissociation constant. Okay, good. Okay, so let's take this further. Now this is something I'm not going to go into, but for your own interest you need to sit and solve it. Okay, I'm not going to describe this. You need to derive the Michaelis-Menten equation and so on and so forth. What you need to understand, which is important from your point of view, is this part. Okay, this is what you are asked in your exam. So this is your equation. This is what, so I'm just going to explain all this to you in terms of terminology. Okay, all this essentially leads to this equation. All right, so V0 is the velocity that is initial, max, S is substrate, Km value anyway you know that, and S again substrate. Okay, so what would happen in these given scenarios? This is what is asked of you. Okay, if substrate is much smaller than Km, as you can see, you can ignore it over here, so you have a different definition altogether. Okay, similarly, if S is much greater, V0 is Vmax, and if S is Km, then you have V0 being Vmax upon 2. So now you see you have another definition of Km. So not only Km is here, as you can see, it's a mathematical expression of rate constants. It is also the substrate at which produces half maximal velocity. You get it? Again, Km over here. Okay, so when you are asked to write a short note on Km, what we are really expecting of you is definition and all these other applications. So it's very, very important to understand Km. Okay, so this is another way of expressing Km. Okay, this is its relevance. Okay, so if you ask me what is the definition of Km, it is a expression of rate constants. What are its applications? One of them is it's the substrate concentration which produces half maximal velocity. All right. Okay, we'll come to this second part a little later when I show you the graph. All right. Now, please take a look at this graph very carefully. This is what is important from your exam point of view also. Okay. So, like I said, it is a plot. Okay. Here the x-axis is their substrate concentration, the molar concentration, and here you have the velocity of the reaction. Okay. So typically you have this kind of hyperbolic curve over here, and as you can see at an initial stage here, okay, there is this very, very rapid rise, initial burst followed by a plateauing out over here. Okay, in other words, enzyme is saturated with the substrate. Okay, on the other hand, this region which is marked is almost linear. Okay, which means substrate concentration and enzyme vary with each other. As you increase the substrate concentration, suddenly you have this nice rise over here. Okay, and after some time it stops making a difference. Okay, so this is an example. Now let me ask you a simple question. What do you think is the problem with this graph? It seems to be fine, right? What do you think can be the problem with a graph like this?
That the velocity of the enzyme is maximum, that we can't extend it indefinitely by
Okay, what else?
We are not taking into account that it is also being formed side by side.
Go ahead, just repeat that.
I was saying, sir, that we are not taking account that product is also being formed side by side in this graph. So
No, actually we are. Remember, the velocity of the reaction is based on that. So in a way we are taking into account the amount of product that is formed in the definition of velocity. Okay, in a way, remember the velocity, right? So we are kind of taking that into account, because if it's maximal, what does Vmax mean? Vmax is basically it's moving at such a fast rate that it's producing every product in a very short amount of time. Okay, what else?
As reaction proceeds, the substrate concentration should decrease, but here the graph is made with the substrate concentration increasing.
Well, okay, no, not really. Think of this graph. Don't think about the situation. The situation can be different. Think what is the problem with this?
Can never be reached.
Vmax can never be reached.
Can never be reached.
So that's a problem with this graph. What difference does it make? No. What's the problem with plotting this graph? You have the data.
Instead of not that huge Vmax,
You all are all going over here. You all are missing a very important part, that the problem is here.
the rate is increasing very fast.
It becomes very right. It becomes very difficult. Enzyme kinetic measurement becomes impossible over here. Okay, it moves so fast. It moves so fast that it's very difficult to understand that from this graph. So what do you think can be done? Forget about the reaction. Let's leave the biology part. That's great. It's happening. But what can be done to solve this problem? Just the graph. Anyone?
velocity.
How can I change this graph?
Experiments.
No, forget the experiments. Forget it. Done. I accept everything that you're saying.
We can take log.
If you take log of this, what will happen? Okay, that's one kind of way. You're getting close actually, but is there any other way? Yeah, you're getting close. There is something else we need to. Yes, I think I can see some of the answers. The answer lies here. You see this? So a better way to create, or rather solve, this particular problem, it's not incorrect, this graph. Okay, there's nothing wrong with this graph. There are certain problems in deriving data from this graph. That's all. So therefore, you have what is called the same thing being represented by the double reciprocal plot or the Line-Weaver-Burke plot. Okay, this is called the double reciprocal plot or the Line-Weaver-Burke plot, which is basically, if you look at this equation, okay, it's basically this one. Okay, this is the double reciprocal. So now this is what you have seen over here in this graph. Now take a look at it in terms of its double reciprocal plot. And what you are going to see is this is the same graph, which you can start measuring stuff. Okay, what do you notice suddenly? It's much easier to start calculating things. All right, you also have intercepts, okay, which are actually numerically feasible. Okay, so that is why, although there is nothing wrong here, but the way of plotting enzyme kinetics is through what is called the double reciprocal plot or the Line-Weaver-Burk plot. There are 3-4 other plots also which are basically variants of this only. Okay, but this is the one that you absolutely have to know. Okay, so now let's take a look at what is this information that we can pull out from this graph. As you can see, 1 by S, fair enough, 1 by V, not a problem, but see suddenly what we are getting. We are getting a slope, we are getting intercepts. Now each of these intercepts is important. Okay, so this intercept gives you 1 by V max, this intercept gives you minus 1 by Km. Okay, so you are getting a lot more information from this, and we are going to be using this to understand enzyme kinetics further. Are we clear so far? What is the problems that are associated with the hyperbolic plot, and why do we kind of turn to the double reciprocal plot? All right, because of some of these reasons. All right. Okay, so fair enough. Then we'll describe that. Then, like I said, we also need to have a few ways to distinguish what are allosteric kinetics. Okay, so like I said, non-Michaelis Menten, the most important difference in allostery involves cooperativity. Cooperativity. What does cooperativity mean? Cooperativity means, please take a look over here. You can have what is called positive cooperativity, you can have negative cooperativity. What it basically means that when the first substrate binds, somehow the Km is changed in a way where suddenly the affinity starts increasing. Okay, binding of the first substrate molecule increases the affinity of the other active sites or the enzyme per se for that particular substrate. Where are we? Oh, wait a minute. Where in this one? Wait a minute. Why are we neglecting? Why are we neglecting S? Wait, in this one? No, we are not neglecting. ये देखो ना, हम कहाँ neglect कर रहे हैं? कहाँ? ये तो है. Where are we neglecting it? Substrate के बिना तो plot ही नहीं करेंगे. We cannot ignore it. It's there. It's very much there. It's just a different way of expressing the same graph. समझ रहे हो? And that is more numerically. It's not an experimental issue. It's a numerical problem. Okay. All right.
The slope is not assessed upon the max.
Which one? Oh yeah. But that's okay. The slope that you'll see. No. That you'll see. That is a numerical derivation of this. And you'll see the use of it when we come to the inhibition. Okay. I will show it to you. ठीक है? Okay.
Sir, by changing the plot, how does it make a difference? At the end, we have to measure the substrate concentration.
Yes. Right. No. You'll be very surprised. See, we don't only have to measure substrate concentration. We are looking at enzyme kinetics. We want to know rate of the reaction. We want to know turnover of the enzyme. We want to know many more things. हमें सिर्फ substrate तो decrease होगा ना. हम substrate भी नहीं measure कर रहे हैं. The rate of the reaction means substrate is being converted into product. आप खुद सोचो. हम substrate तो measure कर ही नहीं रहे. क्योंकि anyway substrate is being converted into product. और वो those parameters to calculate, we need very clear cut numerics. That is why we use this graph, because उसमें हमें पता ही नहीं चलता, क्योंकि where the calculations take place, the kinetics are so fast. Maybe you will not appreciate it over here, but when you actually put it in the laboratory in a test tube, you'll see it is almost impossible to actually put down that data. It moves so fast. So therefore, okay, this strategy was adopted to make some of these derivations a little easier, so that you get more information. Okay, to make others, how are you going to calculate them? Okay, so it's more out of calculation more than anything else.
Okay.
Okay. Okay, chalo. So one is positive binding, the other is negative cooperativity. Alright? Negative cooperativity means the same thing, it's the opposite. If there is a first substrate, it basically decreases the affinity of the enzymes for others. Okay, so you have positive cooperativity, negative cooperativity. Okay, so we are distinguishing between, now in your mind you must be already creating graphs, creating differences between kinetics and allosteric kinetics. So these are the points of difference. Cooperativity, very important component. Allosteric enzymes are almost always seen in what are called multimers or multimeric enzymes. What do you understand by a multimer?
More than one polypeptide.
More than one polypeptide, right? So, and what is in terms of the protein structure, where does this fit? More than one quaternary, right? Is this clear to everybody? For a monomeric enzyme, that means one polypeptide, its top level of organization is the tertiary structure. The moment it involves another polypeptide which also interacts, you go to quaternary structure. Alright, so that's a multimeric enzyme, okay, which has more than one polypeptide. Many of these enzymes which have more than one polypeptide actually come together to form an active site which is at the interface of these molecules. Okay, so it's no longer buried in one protein, it's at the interface of two different polypeptides. Alright. Okay, so again, I won't go into the derivation of this, but for your differences, you at least need to write down the equation in, okay. The standard way of representing allosteric kinetics is what is called the Hill's equation, okay, and basically it means this. If there is positive cooperativity, once you see the equation, you'll know what I'm talking about. I haven't done it over here. And when there's no cooperativity, is n is equal to 1. Important, the plot is sigmoid. This is the plot of an enzyme that exhibits allosteric kinetics. You see the difference? In this case, this was the default plot. Okay, so this is the hyperbole. This is the sigmoid plot of an allosteric enzyme. So as you can see what it means, see the definition, that means it involves positive creativity. What it's saying, first substrate increases the affinity, which means automatically what you're going to see that the rate of the reaction is suddenly shooting up. At very low concentrations of the substrate, over here, there's a very slow reaction. What will happen is that there is positive cooperativity, and suddenly what will happen, the rate of the reaction will shoot up. Okay, and then plateau out. So this is called a sigmoid curve, and it's very, very characteristic of an enzyme or a protein that exhibits allostery. Okay, a very important example. In this case, it's not an enzyme. Anyone knows an example? Yes, yes, yes. It's a very, very important. It's not in terms of enzyme, but yes, in terms of hemoglobin and oxygen, it's more or less synonymous. Okay, so you need to do that. Okay, so that is so much insofar as enzyme kinetics goes. How are we going to benefit by this? Okay, we are going to benefit by this understanding by what is calling adding an inhibitor. Okay, so you have a substrate and you have an enzyme so far. Okay. Okay, so there is a reaction. See, it is going to be sigmoid in technically this way, you know. Okay, okay, so the graph, yes, it will be if you look at it. Now, for instance, if I remove the substrate from here, okay, what it will do is it will start decreasing the affinity for the enzyme and it will go backwards. Okay, so what basically it will do is you can have an enzyme this, and you can have it the other way around also. Okay, it will simply block anything. So yes, you can have that. Okay, you can have, in fact, you have negative allosteric kinetics. There is no Km value. Yeah. Okay, so there is no Km value insofar as that. The computations that are associated with allostery are far more complex, but yeah, there is no Km with that. So in fact, that is for we actually use Michaelis-Menten in many ways to teach you about enzyme kinetics, but in many cases the other one is more applicable. It's more frequent to see allosteri than actually one enzyme, one substrate, and so on and so forth. Right? So that's I think a fair way. Okay, so inhibitory enzyme kinetics. Enzyme, substrate, now we've added one more component. Let's add an inhibitor to this. Okay, when we add an inhibitor to this, it starts doing things. Okay, traditionally an inhibitor, okay, will, when I'm talking of an inhibition to a reaction, what is the parameter that I'm looking at? Out of the stuff that we've already covered, what do you think will be affected when I refer to an inhibitor?
The rate and the affinity of the enzyme for the system.
Affinity, fair enough. The key is the rate, the velocity of the reaction. So when we are dealing with inhibitory kinetics, we are operating at velocities that are suboptimal. All right, it's actually blocking some form of the reaction. So instead of going all the way to Vmax, we are having a problem with reaching that optimal velocity. All right, that is what inhibition is in terms of enzyme kinetics. Okay, now let us say inhibitor was I. Okay, so for all practical purposes, I is either going to bind to the enzyme, in which you have EI complex, or it binds to the enzyme substrate complex, ESI. Okay, what is all this? We'll come to. Okay, traditionally enzyme kinetics or inhibitory kinetics have been divided into two. Those that are irreversible, that means you add an inhibitor to it, that's the end of the enzyme. You know, either it's not going to catalyze it at all, or even more dramatically, it may actually inactivate the enzyme. Okay, and you have something which is relatively more tangible. Those are the reversible reactions. Okay, so like I said, usually what do you understand by irreversibility? So as you can see, usually it involves a covalent bond formation between the active site of the enzyme and the inhibitor. Many of you all have seen these groups, okay, and you all will soon do some part of toxicology and all that, where you will see that many of these functional groups actually make their effect by actually causing irreversible enzymatic inactivation. Okay, so like I said, basically it involves groups which involve hydroxyl, sulfadiol with various amino acids. Okay, so this is irreversible. You can have two types of irreversibility. Okay, what is called truly irreversible, basically for this example, okay, by a covalent modification, or what you have functionally irreversible. Okay, now when I'm saying functionally irreversible, it means that the inhibitor is there, it's binding to it, but it's binding very tightly to it. But if I can find a way of disassociating it, I can get reversibility. Okay, in other words, for that moment it is irreversible, but I can find a way, the enzyme is not really affected. I can still do something to that particular drug molecule which can push it away and give me activity of that particular enzyme. Okay, so you have either irreversible, where there's nothing you can do anymore, okay, and functionally irreversible. All right, that comes to a reversible inhibitory kinetics. We talk about four mainly: competitive, non-competitive, uncompetitive, and what is called mixed. From your understanding of enzymology today, what do you think is the one which is most common?
Competitive, so mixed.
Competitive.
Mixed.
More, I can just 4-5 voices. Some more? Mixed, competitive, yes, let's see what else.
Competitive.
Yeah, I can see competitive. Okay, fair enough.
Non-competitive.
No, the answer is actually mixed. The answer is mixed because, like I told you, a lot of this is involving a protein whose interactions are far more complex. So maybe for the truth is, this is what is the actual scenario when you're dealing with enzyme, substrate and inhibition. And you'll realize that later on that most of these interactions are mixed. There's very few examples which are strictly competitive, strictly non-competitive, okay? But essentially you're dealing with mixed. Again, the differences between these four you must highlight with 6 to 7 points of difference. So you can get a graph, you can get differentiate between competitive, non-competitive, 7 points. Competitive, mixed, 7 points. So what I'm going to describe to you is now some of those headings in which you are going to address these differences. All right? Okay. So this was irreversible. Okay. Now again, just don't get worked up and don't panic. Okay. It's all very straightforward and simple. Remember, we are still dealing with kinetics over here. Let's go stepwise to our favorite equation, right? E plus S, ES, K1, K minus 1, K2, E plus P. Nice? Suddenly we add I to this set. In this case, what's going to happen over here? I is going to bind to the enzyme. Simple enough? So you have Ki and you have a complex with E and I, right? It's an inhibitor. So there's a chance that it will lead to no product formation. Fair enough? That's what our inhibitor definition means, no? That it will not give you a product. So in this case, the inhibitor is binding to the free enzyme. All right? Okay, clear? This is its equation, but for a moment we can ignore this right now. Okay? This looks so much more complex, right? But this isn't. Okay? This is just one step more. Remember, here the I was added to the E. In this, it's the same graph. The only difference is I is added to ES also. Again, very slowly, simple, no problem, right? Inhibitor binds to the enzyme, forms a EI complex, it doesn't do anything. On the other hand, here the I is binding not to the free enzyme, but it's binding to the enzyme substrate complex. Also a Ki for it. Now you're getting ESI. No longer EI, but get it? So EI forms either EIS or ESI. And in this case, okay, you can either have no product formation or some product formation. Okay, so this arrow or this diagram is important to understand this. Okay, so I'm going to go into each in detail. You'll probably understand it, but this is what typically happens. I with E, I with ES. Okay, okay. Now let's see how does this affect. Remember, they're inhibitors. Now let's see what happens. Okay, we are going to talk about the Lineweaver-Burke plot of a competitive inhibitor. Let me explain this graph to you because sometimes you'll are asked to draw this graph. Okay, so the black dotted line is the one which is normally present in the absence of the inhibitor. The activity of the enzyme in the absence of the inhibitor is in black. Okay, when I add the inhibitor, okay, I'm getting the red color. Okay, but as you can see, what it is doing is it changes the graph. It changes the double reciprocal plot of this particular interaction. Let us see what happens in a competitive inhibitor. Okay, now when you're talking about an inhibitor which is competing with an enzyme, okay, and substrate, what are you doing over here? So the assumption is that there is a single active site, there is a substrate. What if the inhibitor was something that looked like a substrate but did not have the functionality, a substrate analog, so to speak? What would it do? What do you think it would do? Loud, no, forget the kinetics, leave the kinetics, we'll deal with that later. What do you think that it'll do? You have a substrate, yes, it will compete with the right, exactly. So because looks like the substrate, it competes with the substrate for the same active site. Now suddenly, what are you going to have in terms of complexes? What is the majority of the complex going to be? You are going to have a mixture of EI and ES, correct? Yes, no?
Yes, sir.
If my inhibitor here is competitive and it's binding to this, I'm going to have a mixture, right? EI and ES, correct? Now EI is obviously not going to do much to the reaction. So what is going to happen in this situation will be, which parameter will get altered?
KM.
The Km will decrease because for the same amount of saturation, more substrate will be required.
But it's an inhibitor, no?
Probability increases.
No, don't get into that complexity. We are dealing with the velocity. Remember, you must talk of the velocity first, because by definition, inhibition means the velocity is suboptimal. You understand my point? By definition of an inhibitor, you are talking of a velocity which is suboptimal. So in this situation, yeah, we'll come to that doubt. I know what you are thinking. Okay, the velocity, overall velocity of that particular kinetic starts dropping, because now there is a mixed population. It's no longer ES, remember? It is only ES that can form the product. EI cannot do anything. So in terms of Vmax, the proportion of the enzyme that is bound to the substrate is less. As a result, the overall velocity of the reaction starts falling. Okay, you remember that? Now, but what will happen to the Km? Think.
It will increase. It will increase.
Km will increase. Why do you think Km will increase? Because
Because now for the same velocity or the substrate, more
Greatly substrate concentration will be required for having.
okay, fine. Now what we are going to, now I'm going to show you how we are going to do this. So this is, okay, so let's put it this way. Okay, so this is our traditional graph. Okay, okay, so this is our traditional graph, what we are going to see. So we have to take into two things over here. We are going to talk of the affinity of the enzyme, and we are going to talk of the velocity. By definition, the velocity has to decrease. Okay, that part is clear. Okay, now what happens? Now see, when I put red over here, okay, what's going to happen over here? You see this red? Okay, I am increasing it over here. Okay, so what's going to happen over here? The intercept remains the same. So in this case, the velocity will decrease, but if we can increase the substrate more and more, we will get equivalent velocity max. In other words, we are pushing out the inhibitor from the substrate, and therefore this type of competitive inhibition is what is called circumventable, or it can be basically overcome by simply increasing the concentration of the substrate. More the concentration of the substrate, it will eventually push it out, push the inhibitor out of the active site, and we can get Vmax, which is essentially the same. So like some of you all pointed out, there will be a difference in the beginning, but as we proceed, we will eventually be able to achieve full Vmax by simply increasing the substrate concentration. Okay, so in this case, the red, which was the inhibitor, and both of them at the end will be exactly the same. So 1 by Vmax and 1 by Vmax plus will remain the same, but what it will do is, as you rightly pointed out, the Km, there will be change in the Km. The Km will increase. So what will happen to 1 by Km?
Decrease.
And minus 1 by Km? Increase. Right. So as you can see over here, do you understand? Yeah. So you see what's happening? It's becoming less and less negative. Is this clear? So this is the graph of a competitive inhibitor. Okay. As you can see, when you have nicely pushed out the inhibitor, you have achieved Vmax. But as you can see, the Km or the minus 1 by Km has come over here. So this type of graph is of a competitor or a classic competitive inhibitor. Clear so far? Okay. I'll come to the specific questions with respect to what changes, but first let me describe the whole thing for you. Okay. Okay. So is this clear?
We came to the conclusion that Km increased by seeing the graph because it increased the slope, so we concluded that Km increased.
We are increased, yes. So what the affinity is affected. Okay. Eventually we are able to deal with the Vmax because they are competing with the same active site. All we can do is simply increase the substrate concentration and eventually we'll push out all the inhibitor. Okay, because the substrate is an analog of the inhibitor. So we can accomplish getting equal Vmax. Okay, but what it will do is, to do that, we'll have to increase the K. Alright. Okay, then we come to the next one. Okay, now take a look at this. This is a double reciprocal plot of a non-competitive inhibitor. Now look at this very carefully. Do you understand what a non-competitive inhibitor is? A non-competitive inhibitor will be an inhibitor which will bind to the enzyme at a site that is not the active site. It in a way is allosteric. So it does not, as you can see, it will bind to the enzyme, but it will not, yes, it is allosteric, it will not affect substrate binding. Are we clear? That means to say which complex will be formed? Right, because in this case, in a non-competitive inhibitor, you can have an enzyme that is simultaneously bound to the substrate as well as the inhibitor. Because the inhibitor is not competing for the same active site, they can both be bound. Okay, but because it's an inhibitor and it is binding at another site, we cannot overcome the deficiency that is caused by a decreased velocity reaction. As a result, what will happen to the Vmax? It will drop, and therefore 1 by Vmax will increase. You see this? Okay, 1 by Vmax will drop. There's no way we can salvage Vmax because the inhibitor is binding to a site that is different. Unlike in competitive, where I simply push in lots of substrate, I can push out the inhibitor. Here I have a situation which I can't do anything because I'm stuck. The substrate can also bind to the enzyme, the inhibitor can also bind at a different site, which means I cannot do anything about my Vmax and it is going to fall. Okay. On the other hand, therefore what will mean? It will mean that 1 by Vmax increases. Is this clear? What will happen to the Km value? So nothing. Do you understand why this is equal to each other?
Because the active site is not blocked.
Because they are not competing. Each has their own independent site. Okay, so it's not really bothering the active site per se. It's just that this complex is doing something to it which is not allowing product formation. You could kind of take it and say it's distorting the active site and so on and so forth. Yeah, but if you just go by classic enzyme kinetics, okay, they are independent. There's no change as such, okay, in the enzyme's affinity for its own substrate. So that remains the same. But Vmax drops, 1 by Vmax rises. So this is the DB plot or LB plot of a non-competitive inhibitor. Okay. This is a graph which is actually the more common graph. It's a double reciprocal plot of a mixed inhibitor, which, as you can see, it's possible, anything is possible. It's a mixture of competitive and non-competitive. You see, you can have both, you can have, it's mixed, and there's actually very little, there's really no pattern in this. Okay, but this is actually what you normally see sometimes. Okay, but what I have for you is something here. Can you think of a graph for this reaction? Think for 2 minutes. Think you have an inhibitor. Think you have this. What is going to happen over here? Km will
be same.
Km will be the same. Okay, what else? No, what's the graph going to be like? So what are you observing over here? You're seeing that this is a very special class of inhibitors that is not binding to the free enzyme. It's binding to the enzyme substrate complex. It's not binding to the free enzyme, it's binding to the enzyme substrate complex. Okay, and therefore there is no product formation. Right, now you're going to see a very nice graph. This is the graph of an uncompetitive inhibitor. Remember this word, uncompetitive and non-competitive are two entirely different things. This is uncompetitive inhibition. Do you understand how this graph has come about? First, let's think of the parameters. Leave the graph alone. What do you think is going to happen in a situation like this with regards to Km and Vmax? First, tell me that. What's going to happen to Vmax? Vmax will decrease. That is clear to everyone? Yes, no? Yes, Vmax will decrease because it is an inhibitor. Fair enough, that we understood. But what do you think will happen to the Km? Km will increase. What do you mean by Km increasing? In terms of enzyme substrate affinity, what will happen if the Km increases? Think. Affinity increases or affinity decreases?
Increase.
Right. So if the Km rises, the affinity of E for X is less or more? Less. If the Km value drops, yes, if the Km value drops, what will happen to the affinity?
Increase.
It will increase. So it is it's just the opposite. High Km values basically means there is poor affinity between the enzyme and the substrate. Low Km values, higher affinity between E and S. Is that clear? But in this case, what is happening? Ye to you found out easily. Vmax is decreasing, 1 by Vmax is increasing. But what's happening over here? What's happening over here? I see the red which has become even less. If red has become less, it means what? What has happened to the Km in this case?
Decrease.
Km has decreased. But isn't that a paradox? Because Km decreasing, right, Km decreasing means good, no? It means that you are actually helping the enzyme bind to its substrate. Yes, no?
So we are actually helping it.
Yes, sir.
You get my point. That is why uncompetitive is a very interesting case. Here the Vmax is decreasing, not a problem. But here, what is happening is that the Km is actually falling, which basically means what an uncompetitive inhibitor is doing is it's actually increasing the affinity of the enzyme substrate complex for each other. Correct? But then why is it an inhibitor?
No product.
Huh?
The product must be formed that much. It is giving, so it is providing an alternate pathway for the reaction to go, so the affinity definitely increases, but still the R product which we desired will not be formed in that amount.
No, but why is the Km falling?
Because it is providing an alternate pathway for the reaction to proceed.
But the reaction is taking place between the enzyme and the substrate. So why is the Km falling?
Just say,
Complex. But sir,
so can't we just say enzyme substrate complex concentration is being decreased by making ESI complex? So the Lee-Chatelier principle says that ES would react more and move on.
Very good. Yes, absolutely. You must remember this reason why Km. So it's a paradox. In spite of it being an inhibitor, it is doing something else. Here, when this inhibitor is binding, it is binding to the ES complex. When it binds to the ES complex, it's pulling out ES to make ESI. What is this doing? It is creating this forward reaction by the law of mass action. It's forcing E to come together with S. Yes? Yes, no? Did you understand that? What the inhibitor is doing is it's forcing the E and S to come together. By itself, it's not doing anything. It's giving a product which doesn't do anything. But what it's doing to the overall reaction is it's pulling out viable ES from the reaction. When this product, which we will talk about later, when this is gone, what do you think will happen to the forward reaction?
Forward reaction will increase.
Yes, so you will have more ES forming. Exactly. So Km Km is decreasing. So that is the paradox. You must understand why these parameters change and what is the example for this. Okay. Okay. So this was about uncompetitive inhibitor. Okay. So like I said, what you are expected is, now see, you have a graph. You know what is to expect. You need to write down about what is being formed. So EI or ESI, whether EI or ESI is active, what are the K values, examples and graph. You must be able to enumerate 6 to 7 points of difference between competitive, non-competitive, uncompetitive and mixed. Okay, you must practice this properly. Alright, so now let's very rapidly go through the rest. So these are reaction mechanisms. We won't talk about this much. You need to read through it. Okay, this is single. I won't talk about this. Okay, so this is what we talked of the extremophiles. I don't think we'll have time to go. This is what you all talked about, right? Okay, thermococcus. So this is the extremophile society which I told you. Okay, it's a very nice and valid society, and it gives you fellowships and all to study these extreme forms of enzymes. Okay, so there's a whole society dedicated to this. Okay, so International Society for Extremophiles. Okay, so look into that. Okay, fine. Then we come to the way in which we are going to talk of regulation. Two differences commonly emerge from this. Okay, I'm going to go through this very quickly. There's not much in this. Okay, a factor that affects enzymatic activity and a factor that regulates enzymatic activity. Okay, a factor that affects it and a factor that regulates it. You need to understand that these are two separate discussions and questions. When we are talking of factors that affect it, we are talking about, in principle, what are the physical factors in and around it that will change the activity. Okay, okay, for instance, let's take temperature and pH. Okay, so this is very quickly, you've probably done all this in your 12th. Okay, so there's this Q10 temperature coefficient. So any change, faster reaction rate, collision frequency, and so on and so forth. Okay, and similarly, hydrogen ion concentrations. The optimal pH is this, but as we have already seen, you can have enzymes that work at extremes of this. Okay, you can have alkaline pHs and acidic pH as well. All right. Okay, so this we will go through. Then of course you have substrate, cofactors, inhibitors. Okay, all pretty much straightforward, which you need to know this. Okay, so what are cofactors? You will have differences between cofactors and coenzymes, which you will be asked. You can talk of metallic cofactors, enzymatic coenzymes. These differences you will need to know this. Okay, very quickly we'll talk of what are factors that regulate enzymatic activity. Traditionally we differentiate it as those that do it short term and long term. Okay, what are short term? Short term can also mean actually immediately. Within a very few seconds you can have enzymes that whose activity is changed or regulated. Okay, so in this you will talk about this factors that regulate enzymatic activity. Yes, for instance in this case, the response to substrate, not the substrate per se. Substrate per se was those that affected enzymatic activity, but how does the enzyme behave when alternate substrates are provided? That is your regulation. Okay, then you have, like we have described, presence of what are called effector molecules. Okay, many of these are small molecules, intermediates, or end products. Okay, I'll just show you a quick. Okay, so this is the way in which very small molecules can actually affect, okay, or regulate enzymatic activity. Okay, either it can do it like this. Okay, the molecule forms a covalent bond with an important amino acid. Okay, I'm going to these are some of the important examples which you will know: glycosylation, methylation, phosphorylation, adenylation. There are lots of others. Okay, I'm only going to talk of phosphorylation to you. Okay, so we are talking about, oh sorry, where are we? Okay, we are going to talk about this one over here. All right. Okay, so we very quickly, what is phosphorylation? Phosphorylation of an enzyme is one of the most important modifications to regulate enzymatic activity on very short time steps. Okay, the enzymes usually what they do is they catalyze a phosphorus bond between the serine residues, the threonine residues, and the tyrosine residues. Okay, like I said, many of these are also there, but these are less common. Okay. Okay, so this I've told you, this helps it do it very quickly. Okay, so basically I'll just show you a quick diagram. This is an enzyme. It has its serine residue. Serine residue, as you'll have all done, it has a hydroxyl group. What this does is a kinase. The kinase is an enzyme that typically adds a phosphorus group to this serine molecule. It creates this phosphorylated serine over here. Okay, the reciprocal reaction is what is called a phosphatase. So a balance between kinase and phosphatases is a very important way of regulating enzymatic activity in the cell on very fast scales. Okay, in terms of its relevance, don't worry about the enzymes right now. As you do metabolism, you will see, we'll go through it in detail. But what is important to understand over here is, as you can see, while the phosphorylation of an enzyme can decrease the activity, it is also possible that phosphorylation can increase and decrease both, and same similarly for dephosphorylation. It can increase it, it can decrease it. This chart you must be able to put down. At the end of metabolism, you are going to be asked this question. Okay, everybody loves this question. Okay, it's a favorite question of all the examiners. Okay, so which are the enzymes whose phosphorylation causes a decrease in activity or a dephosphorylation causes an increase in activity, pay attention to this. Okay, I'm not going to, this is not the topic of our discussion today, it's just to highlight phosphorylation as a regulator. Okay, but you need to do this. All right. So then we have talked of response, we have presence of effector molecules. Removal of end product ensures unidirectional metabolic flux. What does this mean? Sounds big, but what does it mean? Anyone? Somebody just talked about it. Let me give you a clue.
Late chandelier's presence.
The same thing. One of the best ways of creating metabolic pathways in the body is in a strange kind of way. You're also, everything. You're using the product into another reaction. By doing so, you are forcing the creation of a pathway. Okay, and that's how you have these pathways being created. So response, presence, removal of end product. Okay, then comes what is called compartmentalization. It's basically along the same lines as remove the end product, but the difference is basically you can either have a physical segregation. That means certain reactions take place in specific organelles. You can have the cytosol, you can have the mitochondria, you can have the lysosomes, and so on and so forth. So one way of bringing about metabolism is by physically segregating different reactions in different compartments. Okay. On the other hand, another way of regulating enzymatic activity is that you have what are called unique intermediates. The most important part in this is glycolysis and gluconeogenesis. Okay, you will be asked this question and you will need to know the steps as to what makes glycolysis. You need gluconeogenesis because there is some unique intermediates that are present over there. Okay, for now this should just suffice. Okay, similarly you have the ability of enzymes to distinguish between various coenzymes. Okay. All right, so we have response, presence, end product, compartmentalization. Another very important regulation is proenzymes. Many times enzymes are formed in what is called an inactive form. Okay, and they have to be actually activated by one or numerous methods. Otherwise, they might lead to something called, they might break down the tissue that is generated in. Okay, so pepsinogen gets converted into pepsin, trypsinogen, trypsin, and so on and so forth. Many of the molecules itself, proinsulin is already, then it's converted into insulin. Okay, similarly procollagen. Okay, then we come to slowly what is called the long-term mechanisms. Okay, long-term mechanisms essentially mean that there is either new synthesis or breakdown. In other words, so far we have dealt with what is there in the cell at this point of time, right? Here we are talking about something that is going to happen. For instance, de novo synthesis. Something is causing more proteins to be formed. There is more transcription, there is more translation, and therefore more enzymes are being formed. Similarly, you can have the degradation. Okay, degradation can mean something is happening to the protein which is causing it to break down. That can be a more long-term effect of regulating enzymatic activity, and some of the examples are what we've already done. I'm going to give you a very nice example over here to show how it is clinically relevant to understand this concept. Okay. Finally, we have degradation. Okay, it's the same thing. Okay, so synthesis and degradation. Okay, so technically you measure it by basically labeling these amino acids, and you have a couple of pathways. Usually they are ubiquitin dependent and independent pathways. Okay, there are some examples which you will come across soon. Okay, so insulin is hypoglycemic because of these pathways. Okay. Finally, we have, as some of you have pointed out, feedback mechanisms. So that's another way of regulating enzymatic activity. Obviously, you have positive feedback, you can also have negative feedback. Okay, so 6 to 7 points on enzyme being regulated: response to substrate, effector molecules, end product removal, compartmentalization, inactive proteins or enzymes, synthesis, degradation, and feedback. Okay. Now, very nice example I'll give you from the clinical side, where it becomes so important to understand this. Okay. There are some individuals who suffer from a set of diseases called porphyrias. Okay. You won't know about it. Some of you will probably read about it. Now, what tends to happen? Porphyrias, in a way, result basically it's a synthesis of heme. Okay. Now, what these drugs do? Now, many of you will come across these drugs soon. Phenytoin, griseofulvin. They do something very interesting. Okay. They synthesize new protein. They cause an upregulation of transcription and translation, resulting in more new protein being formed. As new protein is formed, it starts, okay, using heme faster and faster. Is this okay? Just a minute. There's somebody saying that the voice is not coming.
It's coming, sir.
It's coming. Yeah, okay, fine. Okay. So what tends to happen is, what will happen is that when you give these drugs, in effect, and as more heme increases, it actually starts accumulating in the skin and gets converted into free radicals which can damage the skin. Okay, it can cause a various set of symptoms and signs which you all will deal with later, but the key is it exacerbates porphyrias, certain porphyrias, when you administer these drugs. Because what these drugs do is, as you can see, they create a situation where heme is removed from the pathway. The moment you remove heme from the pathway through cytochrome P450, okay, cytochrome is a set of monooxygenases, okay, which utilize heme. Okay, so what phenytoin does is increases the transcription and translation of cytochrome P40, which uses heme. Heme is out of the pathway. What do you think will happen to the law of mass action? It will accelerate heme conversion. In a way, it actually exacerbates people who are porphyric. Okay, so this is a very, very important clinical example which you'll need to understand the importance of when you're administering drugs, how important it is to understand the basic biochemistry behind these medications. Okay, so I think I've kept you long enough. I must say that you're a very patient batch of students. Okay, we've been at it for almost 2 hours now. Okay, so I think we'll take a break. Okay, and I think we're done more or less. So probably a little bit is left. Probably the role of metals we can always do at some point in time. Okay, fine. All right. So is there anything else that we need to discuss, or I think we'll just give it a break, no? Okay. And if, like I said, any questions that you have which are still outstanding, just make sure you jot them down. Okay, so that at some point in time we can sit back and just talk about it. Okay. Okay, but the points that I've highlighted are almost very, very important for your examination point of view. Okay. Oh, isoforms and okay. So there's a question over here. Haven't you all done that? I'm not supposed to take that, by the way. But I will just very quickly. I don't know if you will understand that, but I'll very quickly show it to you. Wait, I have something over here. I think gone past it. Wait. Yeah. Okay, so okay, now this is a okay. What I just do it. I'll just introduce you to this topic. Okay, I won't go into details, but isoenzymes and isoforms of an enzyme are related, but they are actually not the same. Okay, they are used interchangeably, but strictly in terms of enzymes, they are not really the same. So when I'm traditionally referring to an isoenzyme, okay, what I'm referring to is that functionally they catalyze the same reaction. Okay, but these are independently coded for by the genome. They catalyze the same reaction. They come from different parts of the genome. Okay, like I said, there is a little bit of a confusion with an isoform of an enzyme and isoenzyme per se. In this definition, usually, as you can see, the definition varies according to this. The origin of isoenzymes can be, okay, so there is this concept in evolutionary biology when some genes are basically duplicated. Okay, or the organism basically undergoes different steps of division, and as a result the genome is duplicated. Now what will happen technically is that the same segment of DNA which is coding for a particular enzyme will get duplicated a couple of times. All right, what that will do is it will basically create different enzymes. Okay, but they will do the same reaction. So that's what he said. They are encoded differently, but they may be catalyzing the same reaction. One of these is what is called duplication. It is possible, and that comes in from the alternate splicing of a single gene. That's when you can have isoforms of an enzyme. Okay, so when I talk of isoenzymes, I am referring to coming from different regions of the genome. Okay, isoforms of an enzyme, which are sometimes used interchangeably, okay, are something which is restricted to a particular enzyme only. That means it's possible that out of 2 copies of the same enzyme, 1 copy happens to be inactive. So out of the 2 isoforms, 1 is inactive, 1 is dominant. Okay, so it is okay, but for you as a definition, you need to know that it's catalyzing the same reaction. They have different physical and structural properties, and they're encoded by the genome differently. Those are isoenzymes. Isoforms of an enzyme will be basically the same enzyme has different variants of it. Okay, same enzyme will have different variants. It's possible that out of 2 enzyme copies that are present in the body, 1 is inactive, 1 might be even more active. You get my point? So that is isoform of an enzyme. Okay, okay, oof, that is so much enzymology for one day.
Thank you so much.
Yeah, okay, so I think the attendance, Sharath, what is the attendance? Any idea? Okay, so then just call her then. Okay, okay, great. Thanks for your attention, and if there is some, which I'm sure you'll have many queries, okay, make sure that you jot it all down and then we can chat about it. Okay, okay, thank you so much.
Thank you.
Yeah, thanks. Just be online. I think there's the attendance also, right?
हाँ मैम, वो अटेंडेंस के लिए डिड यू टेक द पिक्चर? हाँ ओके, तो आई वांटेड टू कंफर्म दैट, बट मेरा फोन हैंग हो गया सडनली, तो ठीक है ना।